Yeah, go ahead.
Hey, Bridge, just letting you know the vehicles have required bottom at this time. If we could go ahead and change the center rotation to the A-frame, and we will set bailout today to due south at 180 degrees.
Good copy. Bailout set at 180 degrees.
Yeah, and we are on bottom.
Yep,
just getting set up.
Yeah. Our center of rotation to A-frame.
And so we're here at the Mona Block, which is northwest of Mona Canyon. And it's a really interesting place in terms of tectonics.
Sure.
Because, as people might know, Puerto Rico sort of lies at a convergent boundary. So it's where one plate is coming in and subducting under the other. The North American plate is coming in and subducting under the Caribbean plate. And Puerto Rico actually is in a zone of sort of diffuse tectonics, and it's a microplate. So there's this whole squishy zone between the subduction zone on one end and Puerto Rico and the Caribbean plate. And Mona Block is quite interesting because it sits right where the Bahamas platform is trying to subduct. The Bahamas platform is 20 kilometers thick of carbonates. Ooh. So as a result, what it's actually doing is as that platform is trying to subduct, it's pushing Mona Block up.
Okay.
Yep. So it's an anomalous, it's an unusual bathymetric high.
So this is something that would have been happening for many millions of years here.
Yeah. Yeah.
That's cool. So do we expect to see some of these carbonates exposed?
I do think we will see some carbonates. We do know that the face that we're on, there is a large fault that is just south of that face, but it sort of seems to peter out before it gets to where we are right here.
All right.
And that's the fault I can't pronounce the name of, but you can.
Septentrional fault?
I'm going to make you say it every single time. But this is also an interesting place because there have been historical earthquakes in this area.
Okay.
Not the super giant ones, but there have been earthquakes here.
So pretty sedimentary here.
Yeah.
Lots of soft bottom, but with some rocks just ahead of us. We're already seeing there some white blobs that we don't know what they are. They might be sponges. Whenever I see something like that, I immediately think of sponges, of course.
I think of rocks, if that makes you feel any better. Yeah.
Yeah, go ahead.
Hi, Bridge. I'd like to request the ship move, please. Range 20 meters, bearing 000 degrees, speed 0.3 knots.
Roger,
I think one of the reasons why it's really important to be exploring around Puerto Rico is that there's a very strong geohazards link. So it's really directly important for people who live on Puerto Rico and on neighboring islands because the risk of earthquakes, because of the risk of some submarine landslides, because of the risk of tsunamis. We really need to understand what's happening in the geology of Puerto Rico and the areas around it.
Sure, and that's very important. And also from a biological point of view, we know that Puerto Rico has local fisheries, small-scale fisheries that is very important in terms of supporting lots of livelihoods. So by what we saw also yesterday in our dive were some of those commercial species, and so we could better characterize the habitats where they occur. So we saw for instance, Ruffy, we saw Alfonsinos, we saw Queen Snapper. All of those are quite relevant species in terms of small-scale fisheries.
And I think we're going to see that for most of the dives that we do around Puerto Rico, that there's going to be a strong biological component and a very interesting geological component for everyone. Something for everyone.
Yeah. And I think one thing that is really exciting about this area is that in a relatively small spatial scale, we find all sorts of different ecosystems, all the way from shallower water reefs to canyons and trough banks and seamounts, and even a trench, which is the deepest trench actually in the Atlantic. So this is quite special. So in a relatively short distance from shore, we have all sorts of ecosystems deep sea ecosystems, and that of course makes this area promotes diversity. So we are very likely to actually to find lots of biological diversity, both fish species, but also those habitat-forming species such as corals and sponges. And we've seen a few of them yesterday, actually. Not in great densities and not great diversity on our dive yesterday, but we did see some of them. We came across a few black corals, a number of glass sponges. Those really seem to be dominant yesterday. Ooh.
So we're still just settling in, but now we're getting a nice close-up view of the sediment.
And actually, Michelle is here giving us also a little bit of history with regards to the Mona name.
Awesome, thank you.
Yeah, bridge move is
Michelle here in
complete.
the chat room is saying that the name of Mona comes from the Taino name of the cacique or the chief of the island of Mona, which was translated by the Spaniards as Isla de Amona, and therefore Isla de la Mona. In addition also to the smaller Monito Island, to the northwest of the larger island of Mona. And so this has nothing to do with the monkeys, which in Spanish are also monos, which actually was my first thought.
I was going to say, we talked about this a couple of days ago.
Yes,
yeah.
exactly. Yeah, and I was not wrong, see, Deb. The white things
They're
are sponges.
Yeah, go ahead.
And they are on what looks like fairly weathered little outcrops.
Roger, I have another ship move range 20
Oh, and actually
meters,
the one that
bearing
we're seeing here
000,
to the lower
speed
right
0
in the
decimal
screen,
3 knots.
we've seen it also yesterday, and that's a Sacocalix possibly species Sacocalix pedunculatus. And these are actually rather quite south records for the species. It's mostly known from higher latitudes in the North Atlantic, all along the Mid-Atlantic Ridge, but all the way into the Arctic and the Nordic Seas, actually.
Pilot, this is Watchlead.
Although there is at least one record.
If possible, could we pop another Niskin down here?
Although there is at least one record of the species off Brazil. And this other one that's coming actually into view is very interesting because it looks, it is also a glass sponge. It reminds me a lot of a Yalonema. However, typically Yalonemas have a stalk, and this one doesn't seem to have one. So we'll have to look a little bit more into it. But before that, we need to log this water sample. Yes, please. And so that is done.
Is there something inside that, Joanna?
No, that's actually part, well, we would have to look, but what you're seeing there, a bit more orangey, might be actually a central axis that the sponge has on the inside.
Oh.
And typically when they are stalked, you will see the spicules of the stalk will make, sort of a rope all the way and through the sponge body, all the way to the apical portion there. That's great. So I'm actually quite intrigued about this species because typically all the Allonemas do have a stalk, and this one is, wow, look at that. And it's also very typical that they have these few oscules on the upper portion, and that's where it's through those openings that the water will exit the sponge. The water will enter the sponge along much smaller pores that are distributed across the sponge surface, and those are called
Nav ship move is
ostia.
complete.
Good copy. Thank you, Bridge.
Sure.
So,
Oh, look at that. Actually, we can see that through that superficial mesh of spicules. Wow, that's beautiful.
so Joanna, some of the
Yes.
scientists in the chat room
Yes.
are asking whether the stalk could just be buried
Yeah,
in the
that's
sediment.
what I'm thinking. Maybe it could be a very small stalk as well.
And so our goal for today, we're at about 1610 meters right now. Our goal is to try to traverse up this slope and get as close as we can to the top. We probably won't achieve the top, but we will at least try in the time that we have. And so one of the things that we might attempt to do is move a little bit more quickly than we would normally do when we tootle around on the seafloor. So we may try to move a little bit so that we can really see things and snap zoom and only stop if something very exciting is happening. Of course, everything is very exciting. And so we can't be absolutely sure, but these do look like heavily weathered outcrops, and they are most likely limestone, just because that's what, or something like limestone. There's some kind of carbonate, because that's what we expect to see above us and higher up the slope.
Yeah, and I was checking here my notes, and there are actually 3 species of Iolonema known from the Caribbean Sea, and that's Iolonema kenti, schmidti, and toxetes.
And so what we're seeing is the seafloor is fairly disturbed. There's a lot
Yes, it
of
looks like there's a lot of bioturbation.
bioturbation, yeah.
So possibly a lot of infauna living here inside the sediment. We can typically have small worms or crustaceans that will stir up the sediment.
Mhm.
Yeah, go ahead.
Hey, Bridge, with your request another ship move, please range 20 meters, bearing 000 degrees, speed 0.3 knots.
Roger. Ship move range of 20 meters, bearing 000, speed 0.
So I'll just note, the geology of and tectonics of Puerto Rico is just fascinating. We were talking earlier about the Mona Canyon, which is south of Mona Block, where we are right now, and someone was asking how it was formed. And the way that it's actually formed is it's an extensional basin. It's pulling apart. And that's because that Bahamas carbonate platform that I was just talking about, it is sort of holding Hispaniola in place.
Oh.
So as it's colliding with Hispaniola, it's sort of holding it in place. But that means that Puerto Rico and the islands to the east of Puerto Rico
Are moving further
they're
away?
moving, they're
Oh.
moving to the east. So they're basically pulling apart from one another. So if you would hold one steady, the other one would be moving off to the right. So it's really cool. And that's what's causing that extension of the Mona Canyon, which is between Hispaniola and Puerto Rico.
All right. And do we know about how fast that
Oh, that's a
pulling
good
apart
question.
is taking place?
I'm not sure. We can try to find out, though.
I imagine that all of these are slow, relatively slow processes, right?
I don't tend to think of them as being super fast.
No.
I think...
Oh, what are we getting into
don't
view
know.
here? That's actually a very nice combination. So I think what's coming into view here, again, they're sponges, of course, but I think the globular one with oscles that are really visible, it's a tetractinellate, could be in the genus Geodia. But the brushy one that we're seeing, those white bushes, are actually carnivorous sponges.
Oh.
Yes, they are. Look at that. And so carnivorous sponges is a group within the family Cladorhizidae, and it's a very interesting group because it has evolved this way of feeding itself that is unlike any other sponges. So typically sponges filter water and extract from the water all the nutrients and food and oxygen that they need to live. However, carnivorous sponges have evolved this other way to feed themselves, and so they even their body plan has changed. So they have all these filaments that we're seeing coming off from those central axes are covered in a particular type of spicule that actually functions like a Velcro. So small crustaceans that come in contact with that will become entangled, and then what the sponge does is that it migrates a number of cells to encapsulate this prey, and then through some chemical processes it just digests it. So it's really, really interesting, and in most of the carnivorous species the aquifer system has been either highly reduced or even totally disappeared. And we think this is an adaptation that has occurred in one single evolutionary event.
Yeah, bridge move is complete.
And that,
Sure.
and so all of the Cladorhizids species share this trait, and we think it's a form of adaptation to a food-poor environment. So we find this typically either in deep sea, or if it's in shallower water, typically they occur inside submerged caves, which to some extent have similar conditions to the deep sea.
When you have a chance, could you do a snap zoom on the rocks? Thank you. If you can, that would be great. I'm just going to try to take a little close-in look at the rocks, too. Just take a look at them while we're starting to move. And so, in answer to your question, Joanna, the Caribbean plate is moving about 2 centimeters to the east with respect to the North American plate. So I don't know if that whole 2 centimeters a year would be the rate of opening of the canyon or not, or whether that would be only, whether that might be the full rate and it's not opening as fast.
Okay.
So, but we know that 2 centimeters a year is really how those two plates, the Caribbean plate and the North American plate, are moving apart.
Okay, that's nice to know.
So these rocks are pretty weathered. They have manganese coating on them. And one of the things that I think we talk about a lot on this expedition and we've talked about on previous expeditions is it's really hard to know what's underneath manganese crust.
Yeah.
So you can very easily say that it's a rock, and you can talk a little bit about what it looks like on the outside, but it's really hard to know. And we've been fooled before on some of our dives where we pick up things that we think are one kind of rock and they turn out to be another because they're completely encased in this manganese coating.
So you'd actually have to collect them and break them open to see exactly what they look like inside,
You
right?
cut them open. Yep, thank you.
Just like we do with sponges to look at their internal characters.
Exactly. And that's one of the protocols for when these rocks are collected and brought back to land. They're cut in half. One half is generally archived. It gets polished, and they make a thin section, which is just a very, very, very thin section of rock, and they can look at it through a microscope and look at what kinds, very thin, like microns thin.
Okay.
And they can see what kind of crystals have been formed that form the rock. So it's really quite interesting.
It's been a long time I had geology classes, actually. I remember we saw some of those slides back in the days, and they were really spectacular, the colors and the different shapes of the crystals, etc.
Yep, they don't look always very exciting when you're looking at them in plain light, but sometimes you can do what's called cross polars, and you polarize the light, and then they're brilliantly colored, and it helps to identify which minerals are there.
So see, so many things that are similar between our jobs, huh?
Looking at microscopes?
Yeah. Looking at things under the microscope.
Looking at things under the microscope. And we're seeing some what look like little ripples, but they're really disorganized and very irregular.
Yeah, and we see the accumulation of some organic material on the seafloor as well on those deeper parts.
So it's hard to know whether any of these rocks might be in place or whether they've all kind of rolled downhill from an outcrop further above. Oh, there was a fish.
There was a fish. That was very fast. Oh, our first scan of the dive.
Where?
There.
Oh, I didn't see it. Now I do.
Yeah. Quite rusty. We had an all sort of huge assemblage of different soda and beer cans yesterday in our dive.
And, you know, one of the things we were talking about yesterday was, you know, how unfortunate it is that people let trash into the ocean. But one of my colleagues suggested that some of the trash that we saw yesterday, some of the anthropogenic waste that we're seeing on the seafloor could have been related to hurricanes. Because there's so much rainfall that it may wash trash out to sea.
Quite far.
You know, which is not what anybody wants, but it could have happened. So some of the things that we're seeing may not have been placed there on purpose.
Yeah.
Nav bridge, move complete.
Good copy. Thank you, Bridge.
Sure.
Ship's on.
I wonder what that is. Looks like a dead sponge there. And some of these rocks are actually quite interesting. They seem to have some sort of layers.
So we're just moving up this slope. We're actually at about 1,587 meters right now. And we're still seeing a fairly heavily sedimented seafloor. Although, if you look at the rocks, they are not necessarily very heavily sedimented.
Not as sediment as we saw yesterday, right?
Right. So it is possible that wherever it is they're coming from, it's happened more recently than not. And what we're probably seeing with these lighter and darker spots on the seafloor is probably some sorting, where some of the heavier
Go ahead.
sediments are being left behind, and the finer grain sediments, are moving with current.
Yeah.
Roger, I have a ship move range of 30 meters, bearing 000, speed 0.3 knots.
I'm waiting for you to find a collection for a biology collection so that I can grab a rock at the same time.
So we just need to find the perfect rock.
That's true.
With the
Or a
perfect biology.
sponge sitting on top of a rock,
Exactly.
that would work too.
Oh, so now I think actually here in the center, I think we have another yellow Nema, but this one has a stalk. So slightly to the left. Pilot, can we have a quick zoom in? Yes. Coming into the center now, there's a stalk with a white body, elongated body.
Yeah.
Yeah. Yeah, on the side, yeah. Thank you. And this would be a more, let's say, traditional looking Hyalonema. But let's have a look.
Mm-hmm. That's okay. Gives us a chance to zoom in on the sediment
Yeah.
too. We're all about multi-purpose here.
Yeah, and that's, I think, what it is, indeed.
So that's the same as the one we were looking at earlier with the
so-
orange top?
Yeah, so the other one was, they're definitely different species, obviously. But yes, this is the Hyalonema genus. So there, as I said, there are different, and here you can see very clearly like these very long spicules that are just twisted around, making this sort of, looks actually like a rope.
Mm-hmm.
And very traditionally as well, we have these zoanthids growing all along this stalk. So this is very common. So
Very cool.
this is the typical Hyalonema.
And then from the seafloor, it looked again like we saw pteropods or shells of some type, that some of this
Yeah.
is biogenic.
That is true. Sea angels. Yeah.
What is that?
Yeah, I'm trying to see some details of the openings, but this is very delicate. I think this must be actually relatively small, this specimen here. That's great. Thank you, Pilot. I'm holding myself a little bit from sampling.
I understand. I was just saying that when you start thinking about it, or when the scientists in the chat room start
Yeah.
thinking about what they might want to collect, that's when I might also reach out and just grab a rock. And the reason would be to understand what the composition of the area is.
Yeah.
It would be great if
So
we could get it further up the slope when they might be in place, but
So let's see if we find some more of these different ones, and if we do, then we may want to collect one of these Hyalonemas.
are there any particular corals that the shore-based scientists are interested in trying to collect today?
Yes, some black corals. We've seen only two yesterday. One of them was the Stichopathus, the coiled one. Remember, we saw lots of zoanthids growing over some sort of coil that we couldn't quite understand what it was because we weren't finding any of the living corals.
Right.
And very much at the end of the dive, we did come across two live Stichopathus. And there was another one that looked very bristly coral, black coral, actually talking of Stichopathus, we have one right there in front of us to the left, this coiled orange coral. And yesterday we also saw this other one that we're not certain which species it might be, the one that had this
Like the two together?
yes, exactly.
Like it looked like two bodies that were growing
Exactly.
into one another, and they were sort of like fan-shaped.
Yeah, exactly that one. Ooh, so some large sponges here.
What is next to that large sponge? It's sort of tall and skinny.
Nav bridge,
You know
move
what I'm talking
complete.
about,
No.
Yeah.
I mean this.
Yeah, that brownish
Yeah,
thing.
I don't know. Well, I think we need to zoom in on that. So the bulby sponge to the bottom. Again, I think it's within order Tetractinellid. bit difficult to say which species or genus it may be. It could be a Stelletta. Okay. Sure.
Thanks.
Thank you. Yes, so this, yeah, most likely either in the genus Geode or genus Stelletta.
That is not the most beautiful sponge you've seen.
No, I can understand that. But what's intriguing me is that structure, this upright structure to the top of it. That is strange. Yes, thank you. And this is a glass sponge again.
Very pretty.
Here, yeah, actually several different species. And you can see actually that how in between some of those spicules surrounding the sponge are loads of other things growing or inhabiting, like small amphipods.
So we probably need to move on a little bit.
Yes, we need.
Yeah.
Which is a pity because that was a beautiful bouquet there.
Oh, look at that.
That is a very large sponge there at the back.
Is that a sponge?
It is.
Wow.
Wow. And that's another Hyalonema, I think. I think it will have a stalk on the...
That'd be wonderful.
Thank you. On the back of it. I wonder, can we turn on the lasers?
Really?
Oh, that is okay. Yeah, they're there.
Oh, okay.
So this is, yeah, it's actually...
It's like 20 centimeters.
Yeah, 20 across. That's a large Hyalonema. Yeah. Oh, it's beautiful. Actually, you can see here how that spicule rope actually projects from the center top of the sponge. We can see still some spicules there.
Very cool.
I'm just curious to understand if it does have a stalk or not. But I don't think it's in the best position, but my guess is that this one will have a stalk. So that's very interesting. Oh, look at that beautiful view of the internal structure of the sponge with these huge channels leading out to the oscules. Fantastic. And we see some little amphipods, I think, living there inside the sponge. Look at that. Thank you. Beautiful view. Yeah, and I think it will have a stalk, only we couldn't see it properly. No.
And so we're just continuing to move up slope. And the idea is that if we move quickly through this area that might be more heavily sedimented, we may get up to an area that perhaps there might be something different to look at. These are very interesting. These look pretty weathered, and they look like they probably have a fairly heavy crust of manganese on them.
And I think we're seeing also here and there, some of these sponge stalks that are left behind after the sponge dies. So the main body typically disaggregates, but that stalk, because they're made of these very long biosilica spicules, they will remain on the sediment, and often a lot of other organisms will attach to those stalks and just live there. So they create this sort of special habitat. Yeah, and you can see here perfectly another one with its typical stalk. It's really pretty.
Yep. We're seeing a little bit more sedimentation on the rocks than we were earlier.
So especially on abyssal plains, where you can find some of these species of Hyalonema, often you will see these stalks spread out and you only find or mostly find a lot of associated organisms on the stalk. So they serve a little bit like islands.
Roger, I have a ship move range of 20 meters bearing 0,
as do the rocks, right? Because they also serve as another place where biota can live.
Exactly. And for a long time, the abyssal plains were perceived, you know, as being these extremely vast, flat areas, only with
nothing.
with nothing or just with soft sediment. But more and more, we are seeing that actually in some of these areas you can find also some rocky outcrops or rocky substrate, and typically you will find a lot of species associated to those. Yeah, and we're having another different
Couple of
glass sponge. Yeah. Coming into view here.
What's in the top right now in the view? Is that just a rock, or is there something on it?
I think there's also probably a skeleton of another sponge.
Did I really just say just a rock? Oh, God.
Yeah,
what happened there?
You're spending way too much time with me, Deb.
I know.
Yeah, can we please have a look at those yellow sponges? Thank you. They're actually rather intriguing. So they seem to be growing on top of something else.
Yeah.
Stalk of, I don't know, doesn't look like a sponge to me, the stalk, the base, so it looks like possibly some coral that was living here a long time ago. Oh, there's a little shaunax there.
A what?
A shaunax, the toadfish.
Oh, the toadfish?
Yeah.
Love the toadfish.
Oh, wow. These yellow sponges are very interesting. They remind me a little bit of a species that we've seen also in the mid-Atlantic ridge, the Ertwigias. Wow. Yes, actually they do look very similar. And up in the mid-Atlantic ridge we came across two color morphs, a white one and a yellow one. And so this would fit the yellow one in that case. But I'm intrigued whether this would actually be the same. And you can see here that part of the sponge is already dead,
right.
So you see the exposed skeleton, whereas the yellow part is the living portion of the sponge.
And in this image, that's about halfway down on the right.
Yeah,
right?
It is, exactly. So the brownish part is that skeleton there. Yeah, and again we see...
Look at those.
There's a little squid lobster living inside of this one, I think. Wow. Yes, this really does look like Ertwigia to me.
And one of the things that we should note for people who are on the line with us today is that the zoom function on the camera on this ROV is incredibly good. It's got 18 times zoom. Yeah. So often we're like, oh, wow, that's pretty big. And then you pull back and turn the lasers on, which are 10 centimeters apart, and you realize you're looking at something that is incredibly tiny on the seafloor.
Yeah. So there also we saw an amphipod living inside of it. Oh,
but look, it's on something else. What is?
Yeah, that's what I'm trying to figure out. This looks like to me, it looks like a coral branch, a coral axis stalk, something that was living here probably a long time ago, because it does seem to have some of that manganese coating as well on it. And the sponge just saw this as a good substrate to settle and grow on. Oh, and there on the back, I think it's a Bryozoan. I was going to say, my instinct was going to say carnivorous sponge, but I don't think it is. I think it's a Bryozoan in this case. Fantastic. It's incredible how in such a small area you see all sorts of organisms growing on top of each other. And what we could also see actually inside of this sponge was some scale worms, and we've seen that very regularly. So if you look there at the left portion, you see that more orangey part. I think that is probably one of those scale worms that lives inside of the sponge. Yeah. Oh wow, this zoom is just incredible. Thank you, Pilot. This is beautiful. Look at that.
And I wanted to mention why we keep talking about the manganese crusts on these rocks. So this is a natural feature. So manganese dissolves out of the seawater. The seawater holds a number of different metals, and when it, over time, in the deep seawater, basically it will precipitate out on top of anything it can find, which is mostly rocks, but sometimes you'll see a coral skeleton that is covered in a manganese crust. And these don't happen very quickly. They normally grow only about a centimeter per million years. So this is, you know, it's certainly, it takes a while for them to grow a significant manganese crust.
Yeah, very long time scales. You geologists work on.
Yeah, we often do.
Yeah. Yeah, I guess that's what we call these subfossil corals, especially when we see these rubble fields.
Happy to.
Coral rubble fields.
All right, so now might be a really good time to introduce the people that are in the control room today.
Oh, yes.
We haven't actually introduced ourselves either, but so why don't we start? We can talk about the front row of the control room,
Yes.
and then we can talk about the back row of the control room, and then a couple of people who are standing in and just watching.
Yeah.
We'll tell you who you are.
That sounds like...
But so, Pilot, could you start?
Of course. So I'm sitting pilot right now. My name's Andrew O'Brien with the Global Foundation for Ocean Exploration. In addition to being a pilot, I'm a software and data engineer. And to my left,
Sitting ROV nav this morning is Chris Wright. I am also with the Global Foundation for Ocean Exploration and also a member of the data team as well.
Sitting co-pilot is Sean Kennison, part of the mechanical team for GFOE.
Good afternoon. This is Brian Doros representing the Global Foundation for Ocean Exploration, sitting in at video control and broadcast.
I'm Art Howard, and I'm on clipping, and I'm also with the Global Foundation for Ocean Exploration.
And I'm Joana Xavier. I'm a researcher at the Interdisciplinary Center for Marine and Environmental Research at the University of Porto in Portugal, and I'm the biology lead here at the EX2206. And to my left,
Deb Blixen. I'm with the National Academies of Sciences, Engineering, and Medicine. And by training, I am a geologist and a hydrothermal vent person in overall. And before I go to Sam to my left, I'm going to also mention that we have two people standing in the back of the control room today. Thomas Morrow, who is one of our expedition coordinators and one of our mapping leading, just depends on which week it is, and our ops officer for the ship, Hunter Brundell.
Yeah, go ahead.
Sam.
And last but not least, Sam Candio. I'm an expedition coordinator for
Roger, I have a ship
Ocean
move
Exploration.
range 20 meters, bearing 000,
And this is a fabulous
speed
scene, I have to add.
0.3 knots.
Good copy. Thank you, Bridge.
yeah, and I think we're coming across again one of those unstocked hyalonemas, which really makes me want to collect one.
That is your right, as long as we have time.
Those are really unusual to me. And as you can imagine, I mean, a lot of the species that we'll be coming across have been described over 100 years ago. And so having the opportunity to actually collect some things, whether they are described or there's also the possibility, of course, for a lot of new species to be found. So this is just a fantastic opportunity to get to see them, how they look in situ, you know, because often they will lose a lot of those characteristics upon collection, and especially in the past when they were collected just with dredges or trawls, they would come aboard pretty much smashed up. So often we don't know exactly how those species look like.
And that's even, you know, we were talking about this in the wet lab one day, which is where we process the samples, that once you bring it aboard, its color may change. If you put it in ethanol, its color may change. They may fall apart from the pressure.
Yes, the texture. Some of them are very brittle and just lose their body shape very quickly.
And so that's why we use a variety of different preserving techniques as well. And the idea is to try to keep them at their ambient seawater temperature as cold as it possibly can until we get them in the lab where we can prep them as soon as we can. But we take different pieces of the biology so that they can be interpreted in different ways.
Exactly. So some of it will go will become voucher specimens from which they can be described. And then we take also usually a small portion that will be for molecular work, so for phallogenetic analysis or genomic analysis.
The rocks aren't quite as exciting. Generally, we let them dry and take pictures of them, and sometimes we might try to take a tiny corner off to see what is inside, especially if it's covered with a manganese coating. But most of the work that we do in identifying rocks, we do on land rather than on the ship.
To me, they're just exciting because quite often they come covered in lots of sponges. So.
Yes, they do. I know. And brittle stars, and what else have we seen? We've seen, is it a small anemone? No.
Small anemones.
So
they're small corals sometimes, hydrocorals. It depends a bit on the rock, but yeah, it's very rarely that we get a rock up that there isn't anything living on it.
Right.
Nav bridge move is complete.
Good copy. Take the bridge.
Sure.
I'm seeing there at a distance to the left also another glass sponge, possibly in the family Pharreidae. But here in front of us, this white sponge, I'm not sure what it is. Yeah, if we can.
Yes, please.
Right center. Yeah, thank you. Okay. Okay, so this is another group of sponges. So I think this one is a demo sponge. Difficult to say even the family, to be honest, on this one.
Mm-hmm.
There's a lot of sponges that look very much alike each other. Oh no, actually now that we're getting closer, this is a glass sponge as well. Because it has this very characteristic mesh at the surface that looks like a lace. So on this case, I think actually it is another glass sponge, but an unusual one. And you see that it is really attached to the rock there. I was trying to see if there was any particular type of spicules there attaching it to the rock, but I couldn't quite see it. But that's okay.
And what is the oval thing that's sort of directly above?
At the back there?
Yeah.
I think that is also actually a sponge, another type. That would be a demo sponge.
And while we're zooming in on that, I just might mention, one thing you can see in these rocks is layering. Mm-hmm. So, which to me is a little bit suggestive that we probably are looking at a carbonate underneath all that manganese crust.
So that's why you wanted to look at that sponge.
Yes, that's exactly it.
Yeah, and we can see. So this is a demo sponge, and we can see actually some papillae protruding from the surface of the sponge, and those would be, some of them might be inhalant and others exhalant. So it will be through those structures that water comes in and gets out of the water.
Yep. I mean, to me, these rocks look mostly like they're still out of place. It's sort of hard to know with some of these very big ones whether or not they are buried in the sediment or whether they, too, have come somewhere further uphill. But what's interesting is that these are much more sedimented than the ones we saw at the base of our dive.
Yeah, they are.
So I don't know if we're looking at, if this is the background and we're looking at sediment on top of it, or if these are rocks that are mostly out of place.
So here again we have some Stichopathis, that black coral that we talked about yesterday. It seems to be very similar to the one we saw yesterday. And I'm not sure what's this brown sediment here. it could very well be a spicule mat. So sometimes when you have an area with a lot of sponges, especially for instance Ertwiggias, well, in general, glass sponges, what we can see is that once they die and all the organic part disintegrates, the spicules will remain on the sediment, and they build what are called spicule mats.
So, Joanna, what's interesting about that, though, is that we're not seeing a huge amount of sponges, but we are seeing, we've actually seen that same thing, that sort of brownish matte looking.
On the rocks.
We've seen a lot of it so far, even on this dive that we haven't been at for very long. So it's interesting that we're seeing the remnants of them, but not necessarily a lot of the live sponges.
Yeah. Actually, we're talking of that. We're just coming across another sponge that looks like it has a relatively large portion of the tissue that is dead. Again, very brownish. Unless those are actually two different sponges now.
Next to each other?
Now that we're turning,
That's okay. While we're doing that, I'm looking at the nice platey rocks to the left.
And you love a platy rock.
Who doesn't love a platy rock? All right, so do we think it's two different sponges?
I'm still deciding.
Or even more than two. It seems like there's a bunch of stuff right there.
There's actually a bunch of different things there. But I'm just now focusing a bit more on that brown sponge. I'll just have a quick zoom on that, because if it is a dead sponge, it's not worth spending too much time on it. But yeah, thank you.
That looks like multiple sponges.
Yes, so there are several sponges here. So we have, again, this Pharrea-like sponge, the white one, roughly one that we're seeing there at the base. We're seeing, again, this yellow Ertwiggia-like. Actually, we have two different species of Pharrea. So the yellow one, it's what looks like Ertwiggia. The roughly white one at the base that also has partly dead tissue, is a Pharreid, and the one that's sticking there at the left, top left, is another species of Pharrea. And indeed, the brown, larger sponge that we're seeing there to the right of this, it is a dead sponge. It's a very large, and we can see the remnants of the skeleton, making these rows of spicules.
Looks kind of like a loofah.
Yeah, it's true. It does.
And then there's more sponges below all of that.
Oh, yes. Encrusting ones on the rock. And encrusting sponges are really, really difficult to identify from images alone. I would say that possibly, I don't know, maybe 60 or 70% of all sponge diversity are these thinly encrusting sponges. But of course, they don't often have very diagnostic characters, external characters that we can use to reliably identify them. There's actually a lot of species to be described yet among those thin crusts.
Very cool.
Thank you. Thank you, Pilot.
And one of the scientists in our chat room, Nolan Barrett, asked quite a while ago, I'm sorry, Nolan, I just saw it now, why is it important to be looking at the geology to understand what the geohazard situation is for people on land? And there are probably many ways to answer this, so I'll try one or two, and other people might want to chime in. From my perspective, the reason why you would be looking at the geology is twofold. You would want to be able to perhaps trace out where faults are, and you might be able to find out how long it's been since they last ruptured, because in some instances, faults on the seafloor can cause things like a landslide that can create a tsunami that can affect people on land. And then, so that would be one reason. The other one would be that if you're collecting rocks as well, you get a better understanding of the context. And so then you can learn more about the history, the geologic history of your area. And that too, again, will help with understanding how it's changed over time, and therefore what the hazards are today versus what they might have been in the past.
Yeah, and also very relevant in terms of understanding diversity and distribution patterns, for instance, and looking at connectivity. So everything is interrelated.
What's that right in the middle?
Yeah, that's what it looks like a stalk of an Hyalonema, but I'm not sure that on top what we have is just the sponge or if there's something else. I think
it looks like a siphonophore sitting on top of something. I'm sure it's not.
I was
but-
thinking of a sea. I don't think it is, but I was first thinking of a sea star munching
Yeah.
on the sponge. But as we've seen a few
Roger, I have a
doing
ship
in
move
previous
of
dives, but
30 meters.
Is that
actually,
actually a sponge?
Speed
Not just a
0.3
sponge.
knots.
yeah,
Nothing
it
is
does. It does look like one of those Ealonimas.
That's beautiful.
Can we,
Do you want a quick zoom on it?
yeah, I actually think I want also to collect it.
So one of the scientists in the chat room
go ahead.
asked that if we collect one of these stalks, if we could try to get it from its base.
Yeah, from the base, yeah. I think they were referring to the unstalked one to see if there is actually a stalk underneath, buried into the sand. Yeah, and you can see here beautifully how these, very, very long spicules are twisted around the central axis to make this sort of a rope.
Beautiful.
And it's with this that they actually anchor themselves to the sediment. Oh, and we have a small brittle star there as well. One zoanthid at least. Yeah, there's actually a few of these small ophiuroids. And it's interesting because in this case the sponge seems to be pretty much contracted, and that's not very common to see. I really wonder why that is. So for this sponge, if we can collect it a bit from the base, just like plucking a nice delicate flower. Yes, exactly. So all of that stalk, so if you can actually try to grab it right at where the base intersects with the sediment, that would be great. There might be a little bit of that stalk also into the sediment, so. It's rather unusual, actually, the body of this one. It's so contracted. And with it, we will probably get some also of these ophiuroids as well. So Chrisma is going to be very happy.
Yep, we heard you.
Okay,
Thanks.
thank you. Do people on shore hear them as well? Okay.
But Joanna, I thought we made a deal that you were going to pick up, we were going to get a rock with our sponge.
Only things that were on a rock.
Yeah, I don't feel like you have held up your part of the bargain here.
Sponge on the Rocks.
There's no sponge on the rocks.
It's a new band name. I promise to select the next one to be on the rock.
I don't believe you anymore.
Just like collecting a flower. Look at that. Oh, look at that. Ooh.
Wow.
It had really long roots there.
Yes. Yes, it did.
Look at that. Fantastic. Just because of this specimen, I think we're going to have a long night in the lab. I'm sure there's going to be lots of different things growing also, or living within those roots.
I don't think that's right. Is that starboard or port? Okay, thank you.
Port inboard.
That's where I think they're aiming for.
Yeah, that's because this that stalk is quite long, isn't it? Yeah, I think you may have to actually turn it on its body and
Shove it
go.
in.
Yeah, shove it the body first. Yeah, like that. Thank you, pilots. That was excellent collection.
That was the port inner bio box.
So yellow Nema. So whether this is going to be one of those 3 species that are known in the area or yet another potentially new species, we don't know. So this is going to be really, I think this is a great collection.
Hyalonema, is that how you pronounce it?
Yellow Nema.
All right. I think we might need to move a little bit faster in the future if we
Yes, I agree.
if we would like to make it anywhere up the slope, because we have only made it about 65 meters up the slope.
Okay, so we still have a long way to go.
Yes, we do.
But this was a great collection.
Oh,
though.
absolutely. I'm not saying that. I'm just saying that it depends on what we would like what we'd like this to be about. You know, if we want to stop and sample, we certainly can. But if we wanted to try to get into some slightly higher elevations, we might need to move a little bit faster than we had been doing. Right now, there's just a shift change going on with the ROV team in the front. So you might hear some murmuring, because they're all telling each other what has been happening on the dive, what they expect that they may see next, what we've already collected and what we might be interested in. So they do a great job of telling each other what's going on so that when the next shift of pilots, co-pilots, and navigators comes in, everyone is fully briefed.
Yeah.
And on a smaller, slightly less noisy scale, that also happens for the video engineers and the video clippers as well.
Yeah. I heard sponge, and that sounds like a great idea.
Whew, one-track mind with you. All sponges all the time.
Well, I'm even starting to appreciate rocks, Deb.
I know, I'm so impressed.
And I was actually checking here some notes as well, and indeed, so the yellow sponge that we saw earlier, that I said, you know, it looks very much like the Twiggia falcifera that we find on the Mid-Atlantic Ridge. It is indeed reported actually for the area. Well, not necessarily here, Puerto Rico, but certainly to the Caribbean Sea. So that's actually very interesting to know, because then we see some sort of connectivity between the area that we're investigating now here and the Mid-Atlantic Ridge and the Azores Plateau where we were previously.
Well, that makes a lot of sense.
Yeah. I'm actually quite excited that we are here because...
Great, thank you.
This really expands, certainly for me, also the knowledge on the sponge fauna of the western and tropical Western Atlantic. And we know that the Caribbean Sea is a biodiversity hotspot, both on land and, you know, at sea, especially in shallow water. And we would expect that the same will be happening in the deep ocean around this area.
Go ahead, Nev.
Pilot, this is Watch Lead.
Roger, I have
two questions for you.
30
One
meters
is
bearing 000
whether...
at .2 knots.
Okay. One is whether you would be all willing to introduce yourselves in just a few minutes. We have quiet time? Perfect. And then the other one would be is that we haven't really made a huge amount of progress moving up the slope yet, and I think we're going to try. So if we could, maybe you can tell us not to zoom on things, and then we can keep moving. Perfect. And so whenever you'd like to start introducing folks up there, would be great.
Absolutely. Good afternoon. This is Brian Doros, representing the Global Foundation for Ocean Exploration, sitting in at Video Control and Broadcast Systems Engineering.
Start over. This is Mark Durbin, sitting navigator, network engineer also, and with the Global Foundation for Ocean Exploration.
And my name is Fernando Aragon. I'm sitting as co-pilot. I'm a mechanical engineer. And in the back we have
Art Howard on clipping, also with Global Foundation for Ocean Exploration.
And I'm Joana Xavier. I'm a researcher at the Interdisciplinary Center for Marine and Environmental Research at the University of Porto in Portugal, and I'm here with ocean exploration.
And this is Deb Glickson. I'm with the National Academies of Sciences, Engineering, and Medicine, and I am a geologist. And I think actually I'll just quickly mention that, again, we're at the Mona Block, which is basically an anomalous high, bathymetric high, because this area is right on the edge of a subduction zone, and the part that is trying to subduct underneath where we are, the Mona High, is a 20-kilometer thick carbonate platform called the Bahamas platform. So basically it's not really wanting to go down, and it's pushing up the structure in front of it. And after I've mentioned that, now, Sam, please introduce yourself.
Hey, this is Sam Candio. I'm an expedition coordinator for NOAA Ocean Exploration.
And perhaps this is actually a good time also for me to make a small summary of where we are in Spanish. So for all of those that are following us from Puerto Rico. Hola, buenas tardes para quien nos sigue desde Puerto Rico. Estamos aquí con la NOAA Ocean Exploration a bordo del navío Okeanos Explorer. Y estamos haciendo la segunda exploración aquí en torno de Puerto Rico. Hoy estamos en el Mona Block, que queda como unas 40 millas náuticas noroeste de Puerto Rico, y el objetivo hoy es hacer una caracterización geológica y biológica de una pared, la pared sur de esta estructura geomorfológica entre los cerca de 1.600 y 1.200 metros de profundidad. Ya hemos podido ver algo de la geología de la zona y también de la biología con algunas esponjas y corales, y hemos hecho un muestreo de una de las esponjas que hemos visto, y ya muestreo también de agua, de la columna de agua y de la agua más cerca del fondo, y eso será para hacer estudios moleculares del ADN ambiental.
RV nav, this is Bridge. We need to get the RVs off the
Muchas
bottom,
gracias
please.
por seguirnos.
Okay, thank you for letting us know. So folks on the line right now, we've had to just come off the bottom right now, and we'll provide you more information as we know.
RV nav, Bridge. Yeah, thanks for getting that moving. Just heard from the engineers we had a sea suction alarm, so they're trying to get the sea strainers cleared. Currently still have DP, but it's possible that we start getting some other alarms, so getting to a safe spot while we get that shifted puts us in a good spot. Will do. Thank you much.
So for those of you who are on shore, we have had to come off the bottom by just about 100 meters. That's what we're aiming for. And they're just troubleshooting an issue with the ship. And once we have resolved that issue, hopefully we will be able to continue the dive. I invite you to take a look at camera 2 if you'd like to see something slightly more interesting than blue water. You can see the D2 from the view of Sirius, which is always a beautiful view to check out on camera 2. So one of the other things that's been very interesting about the monoblock so far was that there has been some exploration on it, but it was back in the 70s and 80s mostly, so a fairly long time ago. We have had a few dives in a few of the areas since, but most of the original knowledge of monoblock comes from dredges, which is basically when you drag a bag on the seafloor and pick up rocks, and you don't really know exactly where they're coming from, and you don't know if they're in place or if they were already tailless. It is a great way to collect a lot of rocks at once, but now we tend to try to know exactly where our rocks are coming from, and that's why an ROV or a manned submersible is a much easier and better way to grab onto those rocks. I will note that I was reading a paper this morning that referenced my undergraduate advisor, Mike Perfit, at University of Washington, who evidently did some of this original dredging in Puerto Rico on this block. I thought was very cool.
Coming full circle.
Mm-hmm.
Yeah, and I think that's at least as important when it comes to biological sampling as well, to see what it looks like in situ, what it looks like attached, and a lot of the organisms that we pull up, like we talk about glass sponges. I think you can imagine how fragile those are going to be. Or some of these sponges that are anchored to the seafloor. And we really wouldn't have a great idea of what those structures are or what they actually look like in their environment without having that video footage as well. So kind of being able to take that high resolution imagery and couple it with some of the further studies that go on down the line, whether it be DNA or further microscopy. It's just critical to have all of that information.
Yeah, I keep imagining what would have happened to those sponges or corals if they're in a large bag filled with rocks that are being collected on the seafloor. You can imagine that perhaps they wouldn't come up in very good condition.
Yeah, you may not even know if they're there at all. They might just be completely
I can tell you it's
destroyed.
sponge mush.
Sponge mush?
Yes.
Sponge mush.
I've seen some of those collections.
The rocks themselves are not really worse for the wearer to be brought up that way, but what it doesn't give us is the context of where it was collected exactly. It gives us a line of where it was
keep
collected.
diving, or if they have concerns with the rest of the dive. So we will get you an update as soon as we have it.
Okay, roger that.
And have you noticed that actually there's considerably less marine snow here today than what we saw yesterday? And
I did notice that.
Yeah. And I think that's partly because we're much further away from shore than we were yesterday. Yesterday we're just a few miles to the north of San Juan. And the amount of marine snow just was spectacular. We're also shallower though. And here we are a bit deeper.
RVNav bridge. Yes, just got an update from the engineers. We have, they were able to change out the strainers. We have a clean bill of health and are good to continue the dive.
Okay, roger that.
Sounds great. And for awareness, now that we've cleaned out a set of strainers, we do have the other set to shift back to. So if the alarm came back, we have the ability to maintain station for a little bit.
Roger that. Thank you.
Okay, for those of you who have been waiting patiently while we've been a little bit, about 100 meters off bottom, we actually are going back down towards the bottom, and we will let you know when we get there. Maybe we'll see some cool stuff in the midwater while we're going back down again.
Jellies.
I didn't see a jelly.
No, I'm joking.
I mean, we can. It would be nice.
Yeah, we may. Or some ctenophores, or some siphonophores, or pteropods. But we're going actually relatively fast now, so. All right, we're seeing bottom again. And we have here small fish coming into view. I wonder if that's a macro-rid fish. We did not see this yesterday.
So we're just taking a quick look at this
Yeah.
fish. Is it a rat tail?
Yeah, possibly, actually. Macro-rid. Yeah, it is. So we're getting a confirmation here that it is a macro-rid.
Can we just say I
Joanna.
finally identified
Also
a rock
rat tail.
correctly?
Woohoo!
Finally.
High five there.
I think the last thing I tried to identify as a fish was not even alive. So I do wonder if some of these rocks are more in place than some of the others that we saw. It's very hard to tell.
Yeah, Bridge, go ahead. Ready. Roger, I have 20 meters bearing 000 at decimal 2 knots.
I mean, so these rocks are fairly heavily sedimented, even more so than we've seen before. And this little section between these two rocks on the left may be a place where sediment can collect and sort of come down slope a little bit.
Yeah, and I wonder if this fish, to me, it looks like an allosauride. We did not see this, so it's actually very interesting because we're seeing the fish we're seeing today are very different from the ones we saw yesterday in comparatively shallower water.
Yep, that is a halosaur.
Yeah. Oh, look at that.
And Joanna, our fish biologist friends often ask for a zoom in near the head. Do you know why that is?
Well, I think it's in general, we should look as much as we can to different parts of the fish, looking at the fins. For instance, sometimes the number of rays on the fins are also very important, but also, you know, on the position of the eyes, the way of the rostrum of the fish looks like. I think all of those are important characters for identification. Sometimes the rows, number of rows of the scales that they have in different portions of the body. So actually, fish taxonomy, it's they have quite a lot of characters that they can look into.
I wonder what's going on with this rock on the far right-hand side. It looks like it's cemented to another rock.
Yeah.
That is one big, massive piece of limestone. But that's really interesting. I'm not sure what that is.
Yeah. It looks like it has a flap.
Yeah.
And Sirius actually gives us a very nice
view
as well.
And it's
go ahead.
Oh, I was just going to say it's interesting because we can see that there are layers in these rocks, but what we're seeing is that they're at an angle.
Yeah.
So that's not how they were originally deposited, right? We know that generally sedimentary rocks are deposited mostly horizontally. They sometimes have cross bedding, but if you're seeing something that's tilted, it looks like this is sort of tilted on its side.
Yeah, we have here again one, I wonder if this is one of the stichopathas as we've seen earlier. Yeah, that would be great. Thank you.
And it just happens to be near a more resistant ridge of that rock.
Yeah. So yeah, so to me this looks rather similar to the one we've seen earlier on this dive, but also yesterday we've seen some, although this one is not as coiled as the one we saw yesterday. So the same general color. The same general organization of those tentacles coming across from the coral axis, the central axis. But the ones we saw yesterday, they were far more coiled.
Aphanostichopathis.
Oh, so maybe it is not a Stichopathus, but another genus related. Oh, so what we're hearing here from Scott Franz in the chat room, that it's what they used to call Stichopathus. Okay, so I found a Stichopathus. So perhaps this is actually different from what we saw yesterday. Thanks, Scott.
So these rocks are all partially buried in sediment, but they do appear to have, they seem to be going, they look like they're going down slope, and that's not how they would have been emplaced originally. I just love that you can still see the layering in all this rock, even though it's all covered in a manganese crust.
Roger, I have another 20 meters bearing 000 at decimal 2 knots.
Okay, and what we're hearing here in the chat is that at this depth, typically it would be an Aphanostichopathus, and then in shallow water, as we've seen yesterday, would be the Stichopathus. Oh, we have here one of those goniasterid sea stars.
The cookie cutter star?
The cookie cutter, yeah.
Or the ravioli star?
Yes, exactly.
And look at what it's sitting on, though, Joanna. Look at how, that looks like biogenic material to me.
Yes,
that does not look like sediment.
So that actually looks like a big mix of with spicules, probably, spicule mat. I see there's probably some leftovers of some Bryozoan colonies as well, and lots of pteropod shells. Oh, look at that. So this is probably a Peltaster. Let's check if we have Chris Ma in the chat room here with us.
He wrote starfish. Come on, Chris, you can do better than that.
I remember seeing previously we had both Peltaster and Blindthaster. And to be honest, I don't know the difference between the two. Yeah, so Chris is confirming possibly Peltaster. The ravioli sea star. Ooh. What is this large sponge coming into view here? Huh.
Go ahead, Neff. Thanks. I think we had talked about doing an extended dive. Is that still on the table? Okay, we'll get back to you as soon as we can.
And so this is also a glass sponge that is here. It actually looks like a very large or very tall glass sponge. It reminds me a bit of the genus Asconema that we find in the Northeast Atlantic. But somehow the body wall seems to be thicker than I would expect. So I'm not 100% sure, so I'm actually checking here some notes as to whether this genus would be expected here in this area. So that's quite interesting, because the only Asconema species that is reported here for the Caribbean is actually Asconema foliata. And that's definitely not this species, because Asconema foliata is a much more, it's much shorter and much more roughly type of sponge. So not at all like this one that we're seeing now. So it's actually unclear to me if this would be in that genus, then which species it would be. We know there are several different Asconema species known from the mid-Atlantic ridge, many of which we don't know exactly how they look like in situ.
Okay, thank you. So one of the things that we noticed before on the rocks, but I was chatting with the navigator, so I didn't get a chance to say it, is some of those layers were vertical. Did you happen to see that? That
on the
where you were, where the sponge was, those rocks were standing up, and the layering was vertical.
Oh, I actually did not know.
Yeah,
I did not notice that.
So you could infer from that that is not an in place rock. That rock has probably come from somewhere else.
And that is an unusual
What
sponge.
is that you're looking? I was going to ask you if it was a very small elephant on the seafloor.
I tell you, I'm seeing all sorts of different sponges here that I do not recognize from the Northeast Atlantic. So
it looks like an elephant.
Different. Yes,
it looks like an elephant wearing headphones.
RV bridge move is complete.
Am I the only one who sees that? Oh, I am the only one who sees that. Oh. Right. Elephant wearing headphones.
Look at that.
And a black coral.
Can we have a snap zone there? That's a very unusual shape.
Seriously, what is going on there, Joanna?
Yeah, I don't know. I'm trying to make up my mind about. I know it's a demo sponge for sure.
So how come it has 3 anchor points instead of 1? Is that normal for a demo sponge?
Yeah, well, that can happen. It's not so common that we see it like that, but that can certainly happen. The surface of it makes me think two completely different groups, actually. So it makes me think both of a tetractinellid and a rock sponge, but the shape is a bit unusual for that. And it makes me think of an aplosclerid sponge, which would be rather brittle. Yes, please, if we could get lasers.
Is that a tiny little squat lobster?
Yeah, or it's actually an Hermes crab inside of a gastropod shell, I think.
Oh, really?
Yeah.
Oh, yeah, I see it now.
It is, yeah. And we have here a little bit of a Bryozoan as well, and we see there a brittle star hanging on from the base from the sponge cliff, almost.
Okay, even the chat scientists agree with me that this looks like an elephant.
This is really weird sponge, weirdly shaped.
Could it have been separate sponges that merged together? Does that happen?
It could. It can happen. Yes.
That was a question from one of our scientists in the chat.
It can happen. Yes, and it has been seen actually also or shown from using some molecular tools that some sponges, especially some of the encrusting ones, can form what we call chimeras. So you look at at a patch sponge, and if we sample different portions of that patch, we can find different genotypes, which indicate that in the past they were actually separated and then they somehow just merged. We don't know exactly the mechanism behind it, why it happens, but it has been shown.
Very cool.
And so these ones that we're seeing now, these are Sacocalix pedunculatus. We've seen those also yesterday, those stalked ones with a nice bulby body, very roughly as well. Whereas these ones here, stalked ones to the right, are Pharea.
Roger, 20 meters, bearing 000 at decimal 2 knots.
And the Sirius view is very interesting right now, and that's the view that is part of the upper part of the two-body system, and so it's looking down on the Deep Discoverer ROV, and you can just see what almost looks like a concentric circle of layers of rocks. I have no idea what that would be. Weird, isn't it?
Yeah.
Yeah.
Looks like they were placed there.
Yeah. Sure they weren't. Well, I mean, they may have been placed there, but...
Somehow,
yeah.
But it's true.
And look at that sponge. It's in what I would have called as totally the wrong place because it looks like it's at the bottom of a sediment chute. So sediment's coming down, and yet that sponge is sitting right in the middle of it all.
Yeah, but that's because that sponge has those particular spicules that make this, sort of root structure, and so allows them to root themselves to the soft sediment. And this is, again, a Yalonima, really nice one.
I just wouldn't think that being in a place where the sediment was actively coming down would be the best option.
Well, but it won't necessarily affect them. Once they've root themselves, they will
pretty
be
stable.
Yeah, pretty stable there. As you've seen when we collected the previous one, the root was still actually quite long beneath the substrate. And as long as the sediment is not falling over the main body where all the canals and openings are, that should be fine. Because that's actually one of the most problematic things with sponges is if they get that aquifer system clogged by sediment.
Look at these giant blocks of rocks, many of which have this layering that you can still see. Almost all of them are completely coated in manganese crust. Many of them have a lot of sediment on them as well. And the orientations are sort of all over the place. So some of them are flat lying when we're looking at them. Some of them are tilted to the right. And so that would generally, we make a wild guess that these are probably not in place the way that, and that they're instead, or that they're in place but they have been deformed and tilted.
And again, we have here another one of those small assemblages of Ertwigia plus Pharea overgrowing something that we couldn't quite make what it was, probably a coral skeleton.
And there is a lot of stuff in the sediment that I would immediately say, that's not a rock. Is that biogenic? Is there something?
I don't know.
Is there something
looks
else?
Actually, yeah, that looks
Doesn't look like a
weird.
rock. Yeah. We're looking at the what's coming into the lower left right now. The thing covered in sediment that's a little...
Yeah, some structure. You see some...
Not rock looking.
Yeah. It's actually
bottom left.
To the left on that small...
It's okay if you can't get to it, though.
Yeah, that's crevice there. Yeah.
I don't know what that is.
I also don't know what that is. Looks like a flow of some sort, but...
That's okay. It gave us a great view of these rocks. Look at that.
Yeah, look at the layering there.
Yep.
To the left.
It's like nearly vertical. That is what we were talking about.
It is that.
That has to be biological. It looks like something got buried.
Harvey Nab Bridge, move is complete.
That is weird, actually. So to the left, it does look like a sponge skeleton, actually a glass sponge skeleton. But these layers, and it's very thick as well. It's rather unusual.
Oh, look at that.
It almost looks like some sort of fabric, but
yeah.
But no, I could recognize the structure of a glass sponge there.
Yeah.
Thank you.
That's really interesting because we see these very thin layers off to the left, and then underneath it, it looks more massive, more blocky. You kind of lose that thin layering structure.
Yep.
But they are all tilting, downward, and that is really very interesting. Thank you so much, pilots.
Okay.
Well, we're certainly making more progress than we were earlier in the dive, so I feel pretty good about it.
Wow.
Yeah.
Look at that.
I know you're saying, wow, look at those beautiful vertical faces on that rock. This is amazing. This is just
It's beautiful. I think there's actually also a Eoplectelli there to the left. Those are these sort of vase sponges that we sometimes see that has this pair of shrimps or a pair of scale worms inside.
And that yellow, the yellow sponge is something we've seen twice now.
Yeah, this is the one, their twig, yeah.
No, Bridge, go ahead.
And then is that a black coral to the left?
To the left, yeah. So a Phanostichopathus. We've been
and so
told
why
there.
is it not Stichopathus anymore?
So that I don't know.
Okay.
I wouldn't know the difference.
Roger, 20 meters bearing 000 at decimal 2 knots.
Certainly something on the morphology of it, probably the internal morphology and the way that also those tentacles and polyps are. I'm just, yeah, again, there's a part, wow.
Oh, that is fantastic.
Yeah, it's a really beautiful view here.
And you can see the difference between, so what these layers must have been, some were more resistant than others, some were, they're probably slightly different in terms of what their composition is and how well they're cemented to one another. And that's how you end up getting layers that are more resistant and less resistant.
Yeah, near to the left is a Bryozoan, I think. Yeah, it looks like a Bryozoan. We've seen one earlier on.
They just look like a tiny tree. Oh, what's the excitement?
Just trying to figure out if it's alive. Yes, I think it is. Yeah, I think
Wow.
it is. Yeah. Really, really delicate. These are also quite difficult to collect because they are very brittle.
So you can barely even see the polyps, right?
Yeah.
It's just, they're like just on the outside of the skeleton.
Yeah, they're very, very... That's great, pilots. Thank you.
Oh, so the question I was asking you, Joanna, whether it's Stichopathes or the other one. So
both
Stichopathes still exists?
It still exists. Only part of the genus has been moved to a Phanostichopathus.
Okay. Get those beautiful layers. It is fantastic. Oh, did you see that giant sponge in the background?
Yeah, there's lots of actually of dead skeletons there as well. And I'm actually thinking if this sort of more lamellar yellow sponge that is to the left of the Ertwigia, bottom left of the Ertwigia, it's actually in a different genus. Remember we saw actually on the mid-Atlantic ridge, we saw a species that grew just like that, normally at the fringe of some of these platy rocks.
Oh, the ones that look like ribbons.
Exactly.
That sort of fringe the...
Exactly.
Yep.
And those were Desmacella, so I was wondering if actually this one that we just saw could be the same thing.
Is that a demo sponge? Or
yeah, that is a demo sponge then.
Look at me picking up all this sponge knowledge.
You are. Wow.
I know, thank you. Very much enjoying it.
And here's again one of these Pharea.
And so all of a sudden... sorry, didn't mean to talk over you.
No, go ahead.
I was just going to say these rocks look a little bit more broken up. You know, we were just seeing these very like large outcrops with long continuous platy layers, and now we're sort of just seeing things that look a little bit more crunched up.
Oh, look at this bouquet here.
That's what you get to all your loved ones, a bouquet of sponges?
It's beautiful. Different species there. Another one of the Sacocalix pedunculatus. This is very interesting because this species, it's not a very common species, but we've seen quite a number of them here.
And so if you look in the D2, in the Sirius camera right now, on camera 2, you can really see this is now, we've sort of hit, you know, a rock wall, and we're going to slowly traverse upwards. Got one hardy black coral going off to the side. It's a little harder to see the layers here, but it does look like there still may be some layers that are kind of coming downward left on the end of the screen, but it's much harder to see.
And look at that beautiful yellow Nima again.
And we're just trying to move a little bit faster than we had been before. So we're at a depth of about 1492, and we're trying to move up this cliff face, while of course stopping to smell the roses and enjoy the sponges and corals.
Can we have a snap zoom on that white stick there, coming the center? Because this is a different coral, I think, unless it's just one that's growing over the stalk of another sponge. Oh, it's a bamboo coral. Yeah, it's a bamboo coral. Oh, look at that. We can see that the skeleton is made of these parts, and there is like these internodes that are darker, so looking very much like a bamboo. So this is actually the first bamboo coral we've seen on the dive, and I don't think we saw any yesterday either. Oh, look at that.
I will plead
Beautiful with
ignorance.
those. We can really see beautiful these polyps details with these 8 tentacles. Okay. We didn't get any info from our shore science team as to whether this is super exciting or not, this bamboo coral. Whether it would be worth collecting. So we're getting at least very good imaging of it.
I'd vote for moving on, personally.
Yeah. And we may come across more on this dive, so.
We could do a collection if scientists want a collection, but we need to know very quickly because we need to stop the ship, which is slowly moving ahead. Wow, look at how much manganese crust is on those rocks. See how they're getting sort of bumpy looking? I think we're going to have to.
We're going to have to.
Yep, thank you. And so one of the reasons that you might hear us asking about collections from the onshore scientists is that we want to make sure there's a reason that we're collecting something. That we're trying to explore connectivity in different ocean basins or different regions. We're trying to look for species that haven't been seen before, or perhaps they were observed 100 years ago.
Exactly.
And we depend on our shoreside scientists to help us determine which samples we should be taking off the seafloor.
Really prioritize, We will not collect just for collecting. We will want to collect with a purpose. And as I was saying, for instance, that Hyalonema that we have collected earlier, the 3 species that are known for the region, they were described in the late 1800s, so that's a long time ago, and now we have a good opportunity. Oh, remember a blue sponge?
Yep. We saw this on one of our first dives.
Yes, so that's very interesting.
In the Azores, on the Azores
Not to say
Plateau.
that this may be the same species, we don't really know, but actually this is a species probably in the genus Hymadesmia, so it's an encrusting sponge in the family Madesmidae. And we've seen a rather blue one as well on the mid-Atlantic ridge. It doesn't necessarily have to be the same sponge. But it's interesting that we find something similar here. So we might be finding actually more similarities than we possibly we could expect between these different faunas. And that's very interesting because typically working on the Azores Plateau or on the Mid-Atlantic Ridge, we tend to compare a lot with the fauna on the northeastern Atlantic, along the European margin. But it's very important that we start having this broader scale view, and especially including both the western but also tropical Atlantic, to try to understand how this... Ooh, what is that coming into view?
Something dead?
This is actually what was possibly...
Several dead somethings?
Can we zoom in on that
Which one?
strange structure to the bottom right?
Yep.
I think this has the same... Okay. This seems to have the same structure that we saw earlier. Remember when we saw that sediment that looked like it was flowing down? And on the side of it, it looked to have this same structure. So I'm wondering what this is. It is... It's very geometric.
And interestingly, what you were saying about connectivity across the Atlantic, that is one of the goals of the ASPIRE campaign, the Atlantic Seafloor Partnership for Integrated Research and Exploration. Understanding the connectivity between the ocean basins within the regions is very important. What is this?
I'm sorry, I'm totally speechless because I have no clue what this is.
Let's see if the handy-dandy chat room knows anybody. Let's see.
It looks dead.
I would agree with that.
It does seem like it has a sponge type of skeleton, but I've certainly have never seen a living sponge that looks like this. But I think it is possibly a glass sponge skeleton. Wow. That is very unusual, and it's also very large.
So let me ask you a question, Joanna.
So it must have a fused skeleton for it to remain like that in such, after being dead, to remain that, cohesiveness of the skeleton. It must have, must be within some groups that have these fused skeletons.
So this is a hypothetical question, but would there be value in collecting a dead sponge skeleton? Could you learn something from it?
Oh, definitely, yeah.
Again, hypothetical question.
No,
but
we could.
Very interesting.
So let's wait and see if we find actually something similar to this, only alive. Because if we do, then we would certainly want to collect that.
Okay.
And that might be what we just saw buried earlier on. When we saw this sediment that looked like it was flowing down and it had like this organic or biological look to it.
And so as we are continuing to move upslope, we just hit another area where we went from having a really rocky substrate to coming back into another sedimented area.
Can we also have a zoom in on those stalk? Yeah.
They're talking up there.
Yeah, if we can have a quick zoom in on those structures there. I suspect they also actually belong to a sponge that lives mostly buried in the sediment. Yes, it is.
Really?
Yes. So this is possibly in the genus Phallogenetic. And they typically, so there will be, buried in the sediment, there will be the main body of the sponge, and it will be like a bulb.
Mm-hmm.
And then they have these long papillae that exit from the sediment, and that basically ensure the flow and the connection to the seawater. Look at those, yeah. Oh, there's a, yeah, look at that brittle star. And there's also a little amphipod crawling there at the left. Yeah, you can see there the detail of these very long papillae. So if we would now dig into the sediment, what we would most likely find is like a, like if it was a potato, like a root, not a root, like a tubercle. The body of the sponge would be lying beneath the sediment.
Not literally a potato.
Not literally
just
a potato.
potato-shaped.
Yeah. And so we are now at roughly
479, 1479.
1479 meters depth.
And we're just continuing upslope. If we move at the pace that we had been moving at, we have a very small chance of making it to the top of the ridge.
Yeah. And so just actually to make a small correction here, that's what I refer to as Phallogenetic. Actually, the current genus name is Oceanapia, so it has been synonymized. In the back of my head, I still have a lot of these old names because of the monographs that I use a lot still on my taxonomic work. But so that would be actually Oceanapia, and there are several species of Oceanapia in the North Atlantic.
Did you say synonymized?
Yes.
What does that mean?
So that means it can happen in various instances, but the most common cases are when you have a species that has been described and assigned to a given genus, and the genus was described, and then someone else comes and describes another genus, but later on we find those to be actually the same thing.
They are synonymous.
And then they are synonymous. And so then the one that was the first to be described will have a priority. So any other genus or species that has been described afterwards that we find to be the same, belonging to the same group, be it either the species or the genus, becomes a junior synonym. And therefore it is no longer used.
That's fascinating.
So it's not uncommon that you see people sometimes referring to the same thing with different different names for the genus, or sometimes even for the species. We try to harmonize this as much as possible, of course, but this is always changing. Or it can change considerably as also we gain new knowledge, both on the morphology but also on the molecular and the phylogenetics of the...
What is the black thing in the center?
I do not know.
Is it a rock covered in something, or is it...
It's a very dark rock, isn't it?
Yeah, it is a rock covered in something. Okay.
It is a rock.
Wow, it is...
But it's really standing out from the rest.
Yeah.
Ooh.
Oh my God, are those all squat lobsters?
Lots of little lobsters there.
How adorable is that?
We have an entire family there. Look at that. There must be something very special about this rock. Huh.
I don't think I've ever seen so many squat lobsters in one place.
No, and they're tiny. So it's like a little nursery there.
Are the lasers on? Right, that's why I was asking. So we're actually zoomed past the lasers right now. That gives you a sense of just how small those
very large.
Lobsters are. Thank you so much.
The lobsters are tiny. Those are maybe some of them 1 centimeter or less.
This is ridiculously cute. It's like a little squat lobster nursery.
Actually, maybe this is not a rock. Maybe this is a piece of wood. It looks like it's very burrowed. Could it be a piece of wood?
Oh, no, that could be. It could still be a rock.
Yeah?
Yeah.
Okay.
We wouldn't know until we brought it aboard.
True.
With its 900 associates. But
no, we don't want to do that.
I could just as easily see this being the same kind of carbonate outcrops that we've seen. But
very interesting.
Yeah, I'm not entirely sure.
At the top.
But there's certainly something very special about this substrate.
And it does look like it's flaking off a bit at the top. Our video engineer just pointed that out. So maybe it is something. Maybe it is wood.
Maybe some wood with some of that manganese coating.
Yeah, no idea.
Yeah, thank you.
So maybe it is a piece of wood.
And we have, again, this bamboo coral that we've seen earlier on. Quite tall.
And we're kind of bumping up against an outcrop of rocks that, if you look in the Sirius view, you can kind of see them moving up the slope.
Yeah. And see, so this is what we refer to. On the bottom right, we can see clearly this is a sponge skeleton again. So the sponges has died, and we see the forming sort of this spicule mat at the bottom right there. So this was probably, again, one of those glass sponges.
That's how we're just moving along and taking a look at this big outcrop.
Yeah, and we see a number of these Hyaloneema stalks also here on the sediment, just without the main body of the sponge.
So we have just under 2 hours left in this dive. And so we still have lots to see. We still have lots of ridge to make up. But I do hope to collect a sponge. Oh, not a sponge. Oh, my God, a
Yes,
rock.
you're
wow,
right.
I'm
You're
doing
right.
so well today. I would like to collect a rock somewhere along this.
With a sponge on it.
With a sponge on it. It's going to be a very sponge-heavy collection cruise, Joanna.
I wonder why. No idea. It's to compensate for all the coral collection cruises.
That's true. There are many cruises that collected a significant quantity of coral. But, you know, as we were saying a little earlier in the dive, if you've just tuned in, we don't just collect samples to collect samples. We collect samples that have a reason, that either they're rare and haven't been seen before, or they are actually characteristic of the site. If you see 20 to 30 of them, maybe it's worth collecting.
Yeah.
Or they show connectivity to another region. So we really want to be thoughtful about how we collect.
Is that again the elephant sponge? It looks like. I really don't know what this sponge is. You don't happen to want that rock underneath it, do you, Deb?
I was looking in the wrong direction, but I feel like it might be too big.
Yeah, no, this is the Bryozoan, yeah, that we're seeing here with a brittle star on it, hanging on to it. Okay, I think the sponge has been definitely nicknamed the elephant sponge.
Somewhere, another sponge taxonomist is very unhappy with you for not knowing exactly what it was.
Yeah, I bet. You know, there are over 9,500 described species of sponges, so...
That's a lot of sponges.
Yeah, and many more to be described still, so... I don't think I can know all of them in my lifetime.
Do you hear that, young people watching? Early career folks? Now is a great time to go into sponge taxonomy.
Plenty opportunity there.
Haven't they found some medicinal properties from pharmaceuticals from sponges?
One of the really cool things about sponges is that there have been a number of people who are working who look at the biomedical properties.
Oh, yes, that is true. There are actually both sponges and corals, but sponges in particular are the single most prolific taxonomic group in terms of chemical marine natural products that have been used for a variety of things. So a lot of those products are antiviral or anti-cancer, antibiotic properties. So they are really sought after for biomedical purposes. And also now more recently there's been a lot of interest in terms also of the biomaterials, so the biosilica that sponges have, also for numerous fields such as tissue engineering. So lots of potential there for in the field of blue biotechnology. There's the
Sponge Wonderland.
It is.
Pilot, this is. I was going to say, Pilot, this is Watch Lead. I was not going to ask to pick up that rock, but I was wondering at another time when we're not actively moving. Oh. Okay. I'm not exactly sure yet, but it would be nice if you could get one of those loose rocks, perhaps the center right at the moment, near the whip coral in front of it, or on top, above it, that's the word. Yeah, is that a good size, do you think?
Like a small one, yeah. Yeah, it's like 10 centimeters, 20.
Oh. That would be fine too. You know, we say that, and then we pull it out of the sediment, and it's 5 times as big.
Yeah. Remember the tooth rock we got once?
That rock was actually much, much bigger than it appeared to be.
Now I'm starting to regret we didn't collect that sponge skeleton, the odd one.
Oh.
That was really unusual.
Well, if we see it again.
Yeah, if we see it again. Okay, then I feel better.
So it was out of reach anyways.
Yeah. Makes me feel much better. Thank you. Ooh. Oh, Deb. I don't think we're taking that one home.
Does not appear so.
No.
Yeah.
They seem to be pretty much welded to the seafloor. Oh. See, and
See how much bigger it is?
It is much bigger.
Objects at your ROV viewing port are bigger than they appear.
And it has a sponge on it.
And it has a sponge on it. Ooh.
Okay, how big is that rock going to be when we bring it up? Okay. Then if we could just spin it, take a couple pictures.
Looks great. Yep.
Oh, it looks fantastic.
Yeah, that could be good, right?
That's not really our question for us.
Okay.
Starboard rock box. At least we know this one won't float away. Oh, shit, it was starboard. Thank you so much, pilot. I think so. I think maybe if we find one more, and it does not have to be here, that exhibits that layering that we've seen, that's small enough to pick up, that would also be great. You're muted. Oh, wow. And we're still slowly but surely making a little bit of upward progress. We're at about 1,460 meters right now.
Pilots, whenever we have an opportunity, can we zoom in on those coiled orange corals? Thank you. Oh, look at that. To me, it looks similar to the one we've seen earlier on. But there's a little bit of discussion going on here in the chat room as to whether this could be a Phanosticopathus possispina. So hopefully with these images, our scientists on shore will be able to determine which species of black coral this is. Yeah, and we can see there at the base the dark skeleton, which is why these are called black corals. Thank you, Pilot. Again, probably a sponge skeleton of some sort. There's a small little sponge there at the front. And there's a little crab there on the back with lots of sediment on it as well.
But it doesn't have that distinctive structure that we had looked at in that earlier sponge. And if you look in the camera 2, the Sirius view, you can just see that we're still moving up a very rocky slope that is heavily sedimented, and you can sort of see some chutes where the sediments are probably being funneled through the rock outcrops. It's a really cool view.
So Debbie, you're getting some support here in the chat room saying that maybe we can get a rock with a bryozoan and sponge on it.
And we should mention to people who are watching.
Yeah, we get 3 for 1.
Exactly. We call those associates. And so when we pick up a rock, often there's something living on it. And so then that would be an associate sample to the main sample of the rock.
Yeah.
However, we had some geo-associates too. We collected a sponge that came on a rock, and the rock became a sample too.
Yeah. And you're the same Alonema, that one that we collected earlier, this very orangey one. And this now really makes me think that these are different species. So we have this more orangey one, and this white one that is coming now in view.
And you suggested that these were rare fauna.
Yeah, but they're not very common, at least the dives we've done earlier.
Right.
Certainly on the Mid-Atlantic Ridge we didn't see any.
But on this we've seen
but here we've
several.
Seen quite a number. Yeah. Both this species and Sacocalix pedunculatus. So this area seems to be very good for these groups.
Very impressed at how fast you can say some of these Latin names.
Oh, many years saying those. Yeah, we need to start finding some common names for sponges. Actually, we don't have that many.
Well, now you have one, the elephant sponge.
Yeah, we have now elephant sponge.
So these rocks look really distinctly different than what we were seeing before. We're not seeing as much of the layering, and they look much more broken up. So the morphology has kind of changed. Oh, but see, here we're seeing some of that layering again at that same angle that's sort of dipping off to the bottom left of our screen.
I wonder if that the right, at a distance, is actually one of those Oceanapias that is partly uncovered. So
the one that had the bulb?
Yes, yes. It reminds me, it looks very much like it.
Pilot, could we zoom into that sponge that's coming in center?
It's not so common that we see them like that, exposed, actually.
Yeah.
So typically, as we saw earlier on, we see those very long papillae, but not the rest of the sponge.
That is really cool, Joanna.
Yeah. So now we get to see the main body of it. So this is most likely an Oceanapia. So that's a Dema sponge. Yes, it is. Oh, it's great. Oh, it has a beautiful surface as well. Never stop to be amazed, actually, with how delicate and how how intricate actually the skeletons of these sponges can be. We have a beautiful ophiuroid here also at the bottom. So I remember, for instance, examining some specimens of Oceanapia in which I had just the bulb or just parts, fragments of the papillae, until we actually realized that these were coming together and they were just broken apart. It's a very nice image. Oh, look. They're perfectly visible, the oscule of the sponge. So it's through that opening that the water exits the sponge. So it will go into the sponge from much smaller openings along the sponge body. It will travel through this aquiferous system. So it's just basically a large network of canals inside the sponge. Several sponge cells will remove from the water the oxygen and nutrients it needs to leave, and then the water keeps flowing out of the sponge. So it keeps this continuous flow of water. So one single sponge can actually filter many thousands of liters of water a day.
A day?
Yeah.
Wow.
It depends a lot on the species, but yeah, it has an impressive filtering capacity. Thank you. Looks like a little sand tornado there. What is that?
Don't know. And I'll just note again that we're in a really geologically interesting area. That this is where one of the parts of where the Caribbean plate and the North American plate are converging together, and the North American plate is subducting underneath the Caribbean plate. And the whole area where Puerto Rico is a diffuse plate boundary zone, including the Mona Block.
Mm-hmm.
And the Mona Block itself is being pushed up out of the seafloor because the Bahamas carbonate platform, which is a 20-kilometer thick package of carbonates, is trying to subduct underneath that area. And it's quite interesting. There's also some evidence that the Mona Block in its lower levels may have the same types of rocks that are found in the Dominican Republic on Hispaniola. So it's just a really interesting area.
So I wonder if technically monoblock is or is not a seamount. Would seamounts be typically of volcanic origin then?
Yeah, I don't think I would ever call it a seamount. When I think of seamounts, I do tend to think of extinct volcanic structures, and that seems to be the general definition. There are probably exceptions to that rule, but I think that's why it's been named the monoblock. It's a piece of the seafloor that is being pushed up and away.
So I guess this is one of those allosaurids.
It does look like
fish that we've seen earlier on.
Oh, not anymore.
Oh, off he goes. And we have here in the coming into center also another one of those stalks of the Hyalonema and another sponge as well. Oh, look at that. So possibly Aldrovandia we're hearing here in the chat room. Thank you.
And that is still a halosaur.
Halosaur. Halosaurid. And that is interesting because this, I remember seeing this fish, and certainly actually within this same genus, earlier this year on another exploration cruise that I had on the Sierra Leone Rise. So that would be just off West Africa, so in the Eastern Atlantic, and we're roughly at the same latitude. I wouldn't say exactly the same latitude, but still subtropical. So it's very interesting because it gives us also some more evidence in terms of the connectivity that might exist across these different subbasins in the North Atlantic. And this is a very beautiful and delicate Pharea sponge. And we can see part of it, or perhaps another one was living here before, and it has died, but the skeleton has remained, so it gets this really dark brown color.
So Joanna, I have a question for you.
Shoot.
I feel like we have seen more dead sponges, or dying may not be the right word, but sponges where part of it is dead and part of it is alive, than we have on some of the previous dives that we've been on this expedition. Is there a reason why there'd be more dead or dying sponges here?
That's a great question. I don't really know. I don't think we have necessarily seen it's mostly because we've seen actually more exactinellid sponges on this dive, and exactinellid sponges because they are more heavily silicified, and their skeletons are more fused than on demo sponges. When they die, we tend to see more of their skeletons because they stay upright. So, and what we've seen in previous dives were not necessarily the same groups that have this more rigid or more fused skeleton. So I think we also notice them more.
That makes sense.
But of course, on the other hand, we think that some of these glass sponges are maybe very long-lived. So some individuals can live hundreds of years. And so it's also quite natural that we will see more of these skeletons being left behind, especially if these communities have been in the region for a long time.
So some of these rock outcrops are much more massive looking. You can see there's no layering in some of them at all.
But interestingly, we're not seeing that many coral species
No.
here.
We've seen more black corals, though.
We've seen more black corals than yesterday, but we haven't seen any of those Clarectinian corals that we've seen yesterday. So yesterday we saw a few species. We think Lophilia or Madrepora. We've seen Dendrophilia. And they were not awfully abundant, but we saw a few, especially in the last part of the dive.
And yesterday was a very interesting dive. It was dive 5 of our expedition, and it was actually an underwater cultural heritage site that we were diving on, with the hopes of finding the aircraft Clipper Endeavour. And we had some great confidence in the targets that we had, because they were very hard reflections off a very soft seafloor. Unfortunately, when we got there, pretty much most of what we found was rocky outcrops that looked not unlike this. It was still quite fascinating, because we saw one that had a huge aggregation of fish.
Yeah, that's true.
And then we were able to wander around the seafloor in a place that we hadn't seen before. So it was very interesting. Unfortunately, we did not meet the objectives of finding the aircraft, but still quite fascinating.
Yeah, and that aggregation of fish had several of those commercial species. So I remember we saw both Alfonsinos, we saw slimeheads, and we saw the queen snapper. They were all actually pretty big. They were not small fish.
No.
Again, there in the center, what seems to be just the papillae of one of those Oceanapias. Oh, starfish.
That's a starfish?
Yes.
Oh, it is. It's a puffy starfish.
Oh. Maybe it's feeding. Maybe it's eating a sponge.
What's interesting is it's not on a hard surface. It's just hanging out in the sediment.
Oh, look at that. That's very inflated. I think it is eating.
It's a slime star, according to Chris Ma.
Slime star.
Pterasterid? I should say that with confidence. Pterasterid. I'm not entirely sure that's how it's pronounced.
Maybe we can have a closer look at the sea star there. And there's a tiny sponge to the top right as well.
We're not looking at the sponge.
I know. Stop talking.
We're looking at this slime star. You can talk all you want, but we are looking at this.
Look at that.
It is really cool.
Oh, it's very pretty. It seems to have like a frill around it.
And as you said, it's very inflated looking.
Yeah, I wonder if it is feeding. Maybe it's munching on something. A sponge, possibly. Oh, yeah, so Chris, you're saying like...
Hymenaster or Pteraster?
Pteraster.
So, Chris, what is the frill around its legs? Arms? It's arms.
Arms. Although here they're not very distinct.
No, they're not really arms.
It's very pentagonal. Calking? I have no idea.
I do not know what that means.
I don't know what that means either. Oh.
Hello.
Hi, Chris.
Hey.
So tell us a bit what we're watching here.
So this is one of the more interesting species of sea stars, the group anyway. And Deb, you were correct. So it's probably Pteraster in the family Pterasteridae. So what you're seeing is really weird, because this family of sea stars has what's called a supradorsal membrane. It's basically like a circus tent that covers over the surface of the actual animal. So the actual proper surface of the star is actually below this.
Oh.
The surface has a whole bunch of rods called pexilla that hold up sort of this extra layer over the top. There's a hole right in the center called the osculum. And in other species, Hymenaster and Pteraster being some of the other more commonly encountered genera, if you provoke this, sometimes they'll produce a lot of mucus or slime.
Okay.
And so, for example, the shallow water Pteraster tessellatus produces so much slime or mucus that you can see it coming off of the animal in big threads. It's considered to be a defensive mechanism. I don't know if all the deep water ones have it, to be honest. I would kind of like to, one of these days, I think I'm going to collect one and have, unfortunately, whoever's on board test and see if it actually produces a defensive mucus, because it'll probably slime everything in the collection box, as well as whoever's. I mean, it's not like it comes out at the high pressure or anything. It's just going to be kind of messy, is all. But that's what that is. The architecture is very odd, and the animals are weird for any number of reasons. But they occur all throughout the world's oceans in the deep sea, and when you get into things like hymenaster, some of the sea stars in this family get to living in the thousands of meters, 5,000 meters or so. So it really is one of those animals that I always adore when I see them, because there's always a little thing to say about them. So,
that's really
but
cool.
Of course, it made me, and they do unfortunately eat sponges. They're pretty commonly. I've never actually seen them observed feeding, but they have, several of them have this large hyaline tooth, which is present on each of the oral plates, and a lot of times, well, there's two things. One, they found gut contents with sponge spicules, but two, they have this big tooth, which is often associated with sponge predation. So, very interesting animals. Oh, and we've got a bamboo coral, so I'll let you get to it. Thanks so much.
Thank you, Chris.
Thank you, Chris. That was great.
So this is when I always like to ask the question of, do we have a stick on the ROV?
Next time then. Thank you. Bye-bye.
Thank you.
Yes.
Bye.
Yes. Yeah, I like the one on the bottom a little bit more than the one on the top, but yep. Excellent. Yeah, exactly.
Oh. Are we getting ready for a rock collection?
We are.
Oh.
Okay. Oh, yeah, that's a really big rock. So, yeah, let's try that. Let's try it and see if maybe pick it up and we can look at it. Oh. Can you spin it around a little bit? Yeah, that looks great. Yes. And will this be going in port? It could go in the same box, so I think we can tell them apart. Okay. Starboard. Mm-hmm. Thank you so much, pilots. Yep, that looks great. I heard that. We might be a little bit heavy on the starboard side because we just put 2 big rocks over on it. And so what we might have to do is to collect another rock that we would put on the port side just to make sure we balance it out. I'm sure we could find another beautiful rock.
Actually, I've seen a rock sponge in the Azores that weighed almost 50 kilos. So
Really?
that was a big sponge, yes.
How big was it, though?
It was like more than 1 meter across.
Wow.
And it being called a rock sponge.
Yep. Heavy. This is really nice, though, because as we're moving up, you can really see the layering in this outcrop.
I'm actually having a deja vu feeling.
Really?
With this rock. Wow.
So now we've finally sort of acquired the wall. Oh, look at that. That is beautiful.
Yeah, look at how those ferrates found that skeleton as a good substrate. And I think we have a small anemone there. Possibly actually the first anemone we've seen in this dive as well.
Whereas I'm looking off to the left, and while there is a sea star there, I was very interested in how the layers are dipping just a little bit. And then every so often there's one that's more resistant, and you can see it sticking out.
Oh, wow. Your eyes are way better than mine. Wow, it's amazing, yeah. I wonder if this is a chrysogorgid coral. Let's wait and see what our scientists in the chat room say. I would not have seen that. That's really impressive, pilot. Oh, beautiful. Yeah, so we're having, it is indeed a chrysogorgia. I think it's also the first one we've seen on this dive.
Mm-hmm. Yeah, I agree with that.
Yeah. I'm still amazed that he saw that from such a distance.
I saw one earlier, but I didn't know that we hadn't seen them before, so about 20 minutes I saw one slip by.
It's incredible how trained these ROV pilots and videographers are.
They are amazing at spotting things that no one else would ever see.
Yeah. I think, is that a I think that's a Metallogorgia. Yes, it is. Beautiful.
Can we take a picture of it and keep moving?
Yeah. Okay. And this is the first Metallogorgia of our dive.
Very pretty.
I don't think we've seen them yesterday.
I don't remember.
The Metallogorgia.
But so Metallogorgia are the ones that always have the brittle star
exactly, coiled
as their associate.
Exactly, coiled into it. But
so pretty.
But remember, we saw them on the Mid-Atlantic Ridge and on the Azores Plateau. So again,
are they fairly common?
Yeah, they don't seem to be uncommon. So I think these are called golden corals. Oh, and you can actually see beautiful the skeleton. It's really looks like gold. Oh, look at that. beautiful skeleton, and can really see that golden color of the main axis, and then these really delicate polyps. So I don't know if this would be also Metallogorgia melanotrichus that we've seen in the Azores Plateau and on the Mid-Atlantic Ridge. There might be different species, but this doesn't seem to be a very uncommon species. At least it seems to have, or a group, it has a very wide distribution range.
Okay.
Oh, look at that color. It really looks like gold. Ah, beautiful. Great shot, pilots. Thank you. So pretty.
So in the off chance we make it to the top of this ridge, Joanna, what do you think we might see there?
I was hoping to see some scleractinian corals. I'm not sure we're going to be lucky, but I was hoping to see a little bit. Well, actually, we're starting to see a little bit more diversity. Few different corals now. So it seems that we might be here at the boundary at which the communities change. I was hoping to see at least one scleractinian coral on this dive. I think we have roughly, 15 minutes left?
Oh, no, we have more than that.
Oh, we have more.
We have about an hour and 15 minutes left.
Oh, okay, that's great.
And I think these rocks are dipping off to the west, if I'm not mistaken, because I think we're looking forward to the north. So these are dipping down to the left, which would be the west. Not all of them.
Oh,
that's fluffy.
Yes, it also has one of these brittle stars, but it looks different from this Metallogorgia we just saw. And somewhat also, I don't know if what we saw earlier, the small one, Chrysogorgia that we saw earlier, it would be the same species as this. The color is quite distinct from the other one. It's very, really white polyps here.
And so we're just continuing to move up slope. We're actually at about 15 meters, so 1,415 meters. And one of our goals for this dive was to try to climb as much of the wall as we could. I think we're doing a pretty good job. We're seeing lots of great stuff. We'll say if we saw another layered rock, I would not say no to collecting it. We're just going to keep moving and looking.
Again, some more of these glass sponges that we've seen earlier, both the Sacocalix and something that looks like Asconema.
What do you think is floating by in the center of the screen right now, Joanna?
It looks like a coral again. Not sure if it's difficult to see. Is it that a black coral? There's again. Oh no, it's
Is it
a
a crinoid?
crinoid actually. Yeah.
Yeah, with the brittle star attached to it.
It is. So very long stalked crinoid there. Yeah. It's so interesting to see all of these brittle stars usually attached to something else, you know, very high up in the water column.
Yep, that's their goal, right?
Yeah.
To get high up in the water
Yeah,
column.
and to get some food. I'm actually thinking if we're in a good position, it would actually be good to collect this sponge that's coming right center to us. So this is what we've been thinking a little bit about, whether it's an Asconema, and the only Asconema that is reported for the region is Asconema foliata, and that's certainly not this species.
I agree.
So I think if we're in a good position to collect, I think we should try. We saw a few of these specimens. I think this is the smallest one we've seen so far, so it would be a good one to collect.
Really?
Yeah. Yeah. So it will be like glass fiber. We can grab it from the bottom, from the base. I think it will rip off relatively easily. It will tear off relatively easy, but it won't be brittle. So it will really feel like woolen felt. So this is actually one specimen that's going to be very interesting to compare. There are a few species of Asconema known for the mid-Atlantic ridge, particularly to the north of the Azores archipelago. So this might very well be either one of those species or something totally new. We don't know. Yes? From the base? Yes. It will tear.
Type in the name. I'll do this.
Perfect. We even got the entire base. It will not sink, possibly. Sure. Looks like there's something also living inside of it. Even though this one is one of the smallest that we've seen, it's still
pretty big,
relatively long. Yeah. Excellent.
Nice.
Great collection. Thank you, pilots. Whew. I always stop reading when we're collecting those sponges because they can be very floating. Great sample. Thank you.
So we just had a very nice sponge collection. And we will slowly continue our way up the slope. And the dive that we're doing today is on what's called the Mona Block, and it's a really interesting area. The whole area where the Mona Block is and all of Puerto Rico and parts of Hispaniola are basically caught between two tectonic plates. So the North American plate is converging, coming together with the Caribbean plate, and that North American plate is sort of diving under the Caribbean plate. But what's happening is there's a whole zone between the North American plate and the Caribbean plate that is acting a little differently than the rigid plates around them. And in the case of the Mona Block, where we are right now, there's something called the Bahamas Carbonate platform. It's a very thick layer of carbonates. It's actually 20 kilometers thick, so it's very, 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 very tall. And that part, the carbonate platform of the Bahamas, is basically trying to dive under where we are right now. It's trying to dive under this part of the Mona Canyon. And what it's doing is pushing up the area that we're in right now. So it's pushing up Mona Block. And it's actually why it's called, it's a why it's called a block. It's just sort of a piece of seafloor that's being pushed upwards. So it's really interesting. And then to our south is the Mona Canyon, and that part is actually pulling apart from, it's extending, it's widening between Hispaniola and Puerto Rico. And that's just one of the interesting geologic features. There's actually very exciting geology all around Puerto Rico. Sorry, that was a miskey.
We're getting here a really nice
Perfect.
view.
Last time I actually wrote it down, so thank you. Thank you for letting me know.
And we're getting here a really nice close-up view of this Hyalonema that we've been seeing along this dive. And if any doubts remained of it being stalked or non-stalked, it's clearly stalked, and we were able to see it here. So it was only one that we've seen earlier on in the dive that didn't look like to be stalked. It seemed like this main body was just sitting on the seafloor. I think we see some tiny amphipods running around inside the sponge. Yeah. Yeah, some of them are inside the sponge, yeah. So they're just swimming around through the canals. Pilots, can we get the lasers on just to have an idea for the size of it? Okay, so it's like, yeah, 15. Perfect. It's like 15 centimeters or 20 across the main body. So these are clearly two different species. So the one that we've collected earlier on this dive is this one that's coming into view now, very orangey and really contracted that we're seeing here. So it seems that we have a few different Hyalonema species just on this area. Another one coming into view. Although interestingly, they all seem to be somewhat different at the apical surface where the oscules are located, which really makes me wonder if we have even more than two species of Hyalonema here. Again, the Hyalonemas are collectively known also as the tulip sponges because they do resemble tulips.
They most certainly do. So this is interesting. We're seeing some more big outcrops.
And again, these layered ones, right?
Yeah, some are layered, some are more massive looking. But all of them are, fairly heavily weathered. They're coated in sediment. Not all. Actually, now, as soon as I say that, then one comes out that isn't really coated in sediment. But yeah, look at these very layered rocks. So see that one on the left? That's the one I really want to get. It's a little big.
It's a little big.
Oh, but what about the one right at its base?
Still a little big.
Still a little big. But that is the texture we'd be looking for.
So do they only break up on these very large blocks?
I don't know.
We can't find these layered ones in smaller sizes?
I don't know. That's a really good question. I'm still very interested by the amount of biological detritus that we're seeing on top of the sediment.
And actually here we see these dark sticks on the sediment, and I'm wondering if those are leftovers of some black corals. We have seen that earlier. I think it was maybe yesterday. Yeah, these black sticks, if we can have a quick zoom in on those. Or maybe some other type of coral just with one of these manganese coating on it. Yeah, actually it looks like more the skeleton of a scleractinian coral, only coated with this manganese crust. Yeah, that's great, thank you. Oh, pilots, can we have a look at that branching white coral there that has also a brittle star on it? Thank you. I'm not sure what this may be, but it seems to have the polyps really nicely arranged, coming from that central axis. One polyp on each side of the central axis. Yeah, so we're having our colleagues here in the chat saying that this is possibly in the genus Narella, so a primnoid coral. So, and that would have been the first one we've seen on the dive. So, indeed, we're starting to see not even if not very abundant, we're starting to see different corals now as we're moving up.
Hi, Joanna. It's Scott here. And
hi, Scott.
Looking at this primnoid, and I was going to say it's interesting because it looks like most of the polyps are coming up in pairs. And just as I was about to say that, I saw one where there was a whorl of 3 polyps. But definitely primnoid, probably genus Narella, as you said.
Oh, excellent. Thank you, Scott. And that would be the first one we've seen on this dive. And I don't recollect that we've seen any actually on yesterday's dive either. Beautiful assemblage there, again with several different glass sponges. It's very interesting because we see a lot this association between the Zertwigia and the Pharea. They seem to come together quite often.
And so one thing that's been really consistent on this whole dive has been the fact that all of the rocks that we've looked at have this manganese crust. And that is basically in the seawater, there are dissolved metals. and those metals precipitate out onto mostly rocks on the seafloor, but we have seen it on corals and, coral skeletons and things like that. And it's really interesting because those crusts contain minerals that can be used in things like modern-day electronics. Oh. So it has cobalt, it has manganese, it has trace elements of other metals. And so it does have a lot of really interesting minerals that are coating these rocks.
Oh, that's very interesting.
Mm-hmm. They form very, very slowly as well. And if you've ever heard of manganese nodules, where it's a small, sort of a small beginning, center, and then it's coated in manganese. The whole rock is coated in manganese. But the problem is that you don't know what's below it. You can't tell from the seafloor what's below it.
Yeah, let's have a zoom in on this sponge assemblage here, which again is growing on top of something else that has died and left this sort of a stalk behind. Actually, this sponge it's also a glass sponge, but it does look different from the one that we collected. Mm-hmm. This looks like a rosellid of some sort, and there's these tiny little sponges coming from the sides of it, and that's actually making me wonder if those are, other juvenile sponges of the same species, or different species of another different species. Wow, look at that. Mm-hmm. There's loads of them.
That's really cool.
And so a lot of sponges, they will produce larvae, and they will have this sort of what we call phallopatric behavior, which means that the larvae will settle in very close proximity, or sometimes even on the parent sponge. Mm-hmm. So this could be one of those cases in which this small larvae is settled on the coating of the parent sponge and has developed into these small juveniles. This is very interesting. It's not very common to make an observation like this. And there are just so many of them. Yeah, and here now to the right we have one of these farreid sponges that we've seen earlier on, mm-hmm. with these tiny amphipods on them. I'm actually, and there's also an hermit crab there. I'm actually seeing if we could find one of these farreid sponges that we could collect, but I'm afraid that if we would try to collect this one in particular, we would probably break off this entire assemblage, and that would be a shame. So let's see if we can find another one of these farreids to use a suction sampler on, actually.
It's a great idea. Those are a lot of little sponges.
Did you look at the white thing at the base of the sponge?
Sorry? Can you repeat that?
Did you look at there's a bright white curling branching thing at the base of this sponge?
Oh, okay.
After you finish looking at it here, maybe
sure.
we can have a look.
Sure. So pilots, before we move on, can we look at a curly thing that is very white that's at the base of the sponge, I've been told? Okay, they're probably there. I think it's probably this. It looks like a brittle star, isn't it? And possibly some bits of bryozoan on a bryozoan. Would that be probably?
Yeah.
Difficult to say.
Yeah, I think it's a bryozoan. That's what I would see.
Right. Okay. Thank you.
All right, we're gonna.
Thank you.
Okay, we're going to keep moving.
Thank you.
And I might talk a little bit more about the manganese crusts if if given the opportunity, unless you would like to talk about that beautiful sponge.
No, just I will keep an eye out for another Pharrea like that one. And if we do come across it, we would like to collect one. We just didn't want to actually, I was afraid that we would destroy that nice assemblage there.
Right. And that's a Metallogorgia,
Yeah, that's a Metallogorgia here, coming into view.
What's the stick-like thing in the top left-hand corner? Are those brittle stars?
Yeah, it looks like a stalk of probably a Metallogorgia, and brittle stars probably are hanging onto it.
Okay. And so we were just talking a little bit about the manganese crust, and as we were saying, what you can't see what's underneath it, but it's really interesting. They do form incredibly slowly, like basically at the millimeter per million years. And the other thing that's really interesting about them is that the crusts only form generally between about 400 meters and 4,000 meters. Deeper than that, you tend to see nodules. So, and the nodules are formed when there's something that you can sort of form around. You know, it sort of starts like a pearl.
Yeah,
it starts
like a
with,
pearl.
You know, it starts with a piece of irritation, and then it becomes a nodule. But these nodules are the crusts are really interesting because they can have things like titanium, nickel, and platinum, as well as manganese and cobalt. So they're often rich in rare earth elements or critical minerals. And those are used in all sorts of products that we use today.
And we just saw another one of those unstalked Hyalonemas. So we do have a number of different glass sponges on this site. This seems to be very good environmental conditions for them, which is, yeah, I have to say, I didn't expect to see these many different types of glass sponges on this dive.
It's been very interesting.
Yeah, very nice.
As are these rocks. Can we find a small one to collect?
Trying to see if this is the same Pharrea that we saw earlier on.
So zoom on the bottom left.
Can we zoom in on that bottom left there? There's like this brown assemblage, but then there's a white sponge sticking up.
Sort of like a candle.
From it, yeah, like a candle. Trying to see if it's the same species that we saw earlier on. We were looking into collecting. Although this one is going to be rather brittle. It's my expectation. So we would need probably to slurp it up, if that would be possible. Can we zoom in first, just to be 100% sure that it is the species that we're looking for? Yes, it is. So if we have, I think this might be best collected by slurping it up.
Right now, I think they need to just stop the ship.
Sure.
While you're looking at that sponge, I'm looking to see if there's anything that we could collect in the background.
Yeah, I think it will be brittle. So possibly slurping it from the top would be the most safe way to make sure we get it. And I think we'll have to we will collect that little brittle star with it. Which number would this be? 07? 07?
No, 08. 8.
Oh, yeah. Okay. Can we try to slurp it from the top? Yeah. Yeah, it's off. Yeah. 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 Got it in the canister? Oh, excellent. Thank you, pilots. That was great. Brilliant. Thank you.
Thank you, pilots. That was beautifully done. Mm-hmm. That's good to know. Thank you. Was it? Okay, so we will check both 1 and 6. Okay. Pilot, this is Watchley.
Pilot, this is Watch Lead. Yeah, we've been swapping folks out for dinner. We're looking for a layered carbonate. If any of these rocks look a little bit grabbable and seem to have some nice layering, those might be a good shot for collection.
Right, so what have I missed, Thomas?
I don't know. I just got in. I just got in right before you did.
Okay.
Not a whole lot. We just
No.
finished up the sample.
Okay.
And then
Brilliant.
pushed off from the rock.
Oh, we're seeing a starfish there. I wonder if this is similar to one we've seen yesterday. Actually, we saw two of these starfish or sea stars. That one was an Ophidiaster and the other one was an Enrisia. Oh, that's excellent. It's a good view of it. We don't have right now Chrisma in the chat room, but we will make sure he gets this image.
Yeah, it looks like the chat seems to think it's an Enrisia.
Okay. Oh, yeah. Yes, and there was also an encrusting sponge there, a very dark one. Are we looking for a rock?
Yes.
Okay.
Deb asked if we could look for a
layered?
layered rock. Yes.
Okay.
The layering is the characteristic that we are after. Okay.
So let's see if we do find one. Wow. Okay. Looks like we have a little dip here now. Oh, is there a roughy? Oh no, it's a Shonax. It's a Shonax. It's the toadfish.
Yeah, you don't really see them.
We don't normally see.
around.
Yeah, exactly. They're
Usually just
normally.
sitting.
Oh, that was his best attempt at a smile.
Yeah, he's a feisty little one.
They're the cutest, no doubt. Look at that. We mostly see them on the seafloor or over a rock.
Just
with
looking
their,
moody.
Yeah, with their grumpy face. But you saw, he tried to
He
smile.
tried. He was putting on his bravest face for us.
Yeah.
He knows it's Friday evening.
Yeah.
Oh, someone here in the chat room is referring to it as a coffin fish. I've not heard of that. Coffin fish. Yeah, but it's very interesting because a lot of fish species have different common names in different areas. So that might very well be. Okay, I think this is again one of these very long
Is that a bamboo?
bamboo coral that we've seen earlier on. And I think I also saw... Yeah, that would be great. Thank you. Not 100% sure if it's the same species as we've seen earlier on this dive.
The chat is requesting some detailed zooms on this coral for a positive ID.
Yeah. So let's see what we can get. This is a little rough area that we're on right now.
Wow. It's a nice view.
Yeah, it is. Against that dark background, you can really see more of the details. It's very pretty. So, yeah, and we can even see the skeleton underneath it with those internodes and nodes, you know, the darker nodes there, which is why they're called these bamboo corals. They really look like bamboo. And beautiful with the very nice. Oh, this is great. Thank you, pilot. Yeah, it seems to be quite long. I wonder, possibly 1.5 meters tall, perhaps. Wow, look at that. And it goes on and on. And that's the base with that basal disc. Great, thank you. This was 8. We haven't found a rock yet for you, Deb.
You haven't found a layered rock?
No.
Oh, that's very sad. I was hoping we might just be able to find one more.
And again, here at the base here of these rocks, we see these dark debris, and we looked a bit, we had a zoom in earlier on, and that actually seemed to be the skeletons of scleractinian corals covered, coated with that manganese crust. It wasn't very clear what the species could have been. Perhaps Madrepora. But truth is that we haven't seen yet any of those scleractinian corals alive here during this dive. Only some of this coral rubble.
And that very dark rock.
Yeah, that's a small sponge there, encrusting sponge. Oh, we also see here a solitary scleractinian coral on the rock at the front of it.
It's interesting.
And there's a Naphanostichopathis.
Yeah.
Oh.
It's just interesting because it has so much less sediment on it than most of the rocks around it.
Do you think it has a different composition
No.
than the other one?
Probably not.
Oh, look. That's a very nice solitary scleractinian coral here. Not sure if maybe a flabellum or... Oh, there's something very bright on it. Oh, that's very nice. There's actually a few things here on this rock. Is that not layered enough for you, Deb?
Not really, no. That wasn't what I was looking for. This is not the rock you're looking for.
Yeah, let's have a look at that. Looks like an anemone.
Is that?
Yes, that is an anemone there. And there's a tiny white coral. That is very small, though, that we're seeing there. Looks like a very small octocoral of some sort, but really difficult to say. This is probably, what, just a few centimeters tall. Yeah, and we see here actually again that those black skeletons on the seafloor. So there must have been other scleractinians, corals living in this area previously.
Well, we have gotten much closer to the top of the ridge, so it's possible it could be coming down slope from above.
Yeah, there's actually quite a lot of it here.
Yes,
right
there really
here,
is.
There's a lot of that coral rubble.
See if only we could just crack off part of one of those little ledges.
Yeah. That's probably not easy, though,
no,
is it?
Probably not.
Oh. So we're now at roughly 1,364 meters depth.
We've got about 20 minutes left on this dive.
So not so much time left anymore. We will certainly not get to the 1,200 meters as we had initially planned.
We tried.
Yeah, and we did some really cool observations.
Go ahead, Nav.
And collections.
Oh, thank you. So So it's actually 15 minutes that we have left on the seafloor.
Okay.
Do you think we can get in one more rock sample before that?
We might.
We have to identify a likely candidate.
I'd be particularly happy if we find one with a sponge or a coral on it. And if not, a sponge will do.
No. Are you ever going to collect a coral?
Well, it doesn't seem that there has been great deal of interest on the ones that we've seen so far. So perhaps they're, either more common or but we never know. We might still come across something really exciting, coral-wise. Ooh, these are very layered.
These are very layered. Is there one that's just a small version of that? Yep. Doesn't look so great to me. What do you think, Joanna? No. Yep. I think that.
Yeah, but it's probably.
I think that one that's in the left center is probably part of the larger outcrop, not separate from it.
It's not going to be loose. Oh, there goes again our Shaunax friend, hiding there.
Oh, okay. Yes. Okay.
Okay.
Thank you. That's okay. So let's look at this beautiful frogfish, which we can only really see its sort of walking feet at its tail at the moment.
We just saw it actually swimming a while ago while you were away.
Oh, sure.
Yeah.
I go to get dinner for 1 minute.
Oh, and that's actually a different coral that's there in view now. Okay.
Okay, thanks very much.
Thank you.
So I was wrong when I provided our 15-minute warning. We actually need to come up. We're going to come off the bottom now. So this was the best view, and what a great view this was of these very layered and platy rocks. And I would say this was an incredibly interesting dive, Joanna.
It was great.
All sorts of things. And so we just wanted to thank everybody on the GFOE team and all of our expedition coordinators on the ship and our mapping folks and all of the shore scientists because they have done an amazing job of making this dive successful.
Yeah, this was really great. It was Glass Sponge Wonderland.
Yep.
It was a great dive, and thank you all very much.